Dr. S. Kupuswamy sir, Director Research, Dr. R. Balagurunathan sir, Director of Skill Development, Mr. M. Kumaravel sir, the Chief Executive, Mr. M. Chokkalingam sir, and Admission Director, Dr. S. Varadaraju sir, in their absentia. I also extend a gracious welcome to all the head of the departments, our esteemed faculty members and dear participants, and our lovable students. We also welcome Dr. G. Kandarajan sir, Scientist, Center for Peninsular Aquatic Genetic Resources, ICAR, National Bureau Office Genetic Resources, Kochi. Dear students and participants, before we proceed further, we have a special video presentation highlighting the journey and contributions of Dr. B.S. Yashwant sir. I kindly request everyone to maintain silence, focus your attention on the screen.
Include cell line development. Gives me immense pleasure and pride. Gives me immense pleasure and pride to warmly welcome Dr. B.S. Yashwant sir, Scientist, C, Center for Marine Living Resources and Ecology, Ministry of Earth Sciences, Government of India, Kochi. Dr. Yashwant sir is a promising scientist in the field of fish biotechnology and marine life sciences. He completed his PhD in fish biotechnology from ICAR, Central Institute of Fisheries Education, Mumbai. Sir's research expertise include cell line development, muscle cell culture, molecular biology, and bioinformatic tools. His master's and doctoral research focused on the development and functional characterization of fish cell lines, especially from Aviparan ocellaris and Labiophyta, which have immense applications in fisheries, biotechnology, and disease diagnostics. Dr. Yashwant sir has successfully developed 7 distinct cell lines for various in vitro life science applications and identified more than 15 unique proteins involved in muscle cell differentiation. These contributions highlight his strong research capabilities and innovative approach. In recognition of his scientific excellence, Sir has received several prestigious awards such as Junior Scientist Award and Young Scientist Award in 2023, All India Best MFSC Thesis Award and Best Oral Presentation Award at an international conference in 2025. He is also an ICAR NET qualified scientist and recipient of ICAR JR Fellowships with 16 research publications, gene sequence submissions to international databases, a patent physical line, and hands-on experience in training and capacity building. Dr. Yashwant sir continues to inspire young minds in fisheries and life sciences. Welcome you, sir. Thank you.
The sea, once it's cast its spell, holds one in its net of wonder forever. With these inspiring words, we now move into the first session of this prestigious national conference on marine resource conservation and valorization of Blue Revolution. Session 1 marks the beginning of our academic voyage today, a session that sets the intellectual tone for all the deliberations that follow. This session focuses on understanding, monitoring, and conserving marine ecosystem using advanced scientific tools which are vital for sustainable development and environment stewardship. We respectfully welcome Dr. Yashwant Sir, BS, Scientist C, Center for Marine Living Resources and Ecology, Ministry of Earth Sciences, Government of India, Kerala. Kindly present your PPT, sir.
Good morning to one and all present here. All my dear participants, students, respected guests, and all the organizers of the event. And very warm morning to everybody. And hope everybody has in the same energy as we are in the yesterday evening. And let us come back to science again for the as part of the conference today. And I think I may not require much introduction. It has already been made from yesterday. A lot of introduction has been made. I am Dr. Yashwant. I am working as a scientist in Ministry of Earth Sciences and currently posted in Center for Marine Living Resource and Ecology, Puduvaipu, Kochi, under Government of India. So actually, I was supposed to talk about the bioactive compounds from the microbes. So suddenly I requested the organizer to change the topic to discuss something new, something unique, something innovative, and something is trending nowadays. And you know, as a biotechnologist or like a microbiologist, we don't want to go to field and we don't want to go to outside our labs. And we wanted to be there in the lab all the time. And when it comes to aquaculture and fisheries, everything happens in the field and ocean. So the idea is, why can't we create the meat in the lab itself? That's what I'm going to talk about today. And before getting to the topic, let me just introduce about my organization and about my ministry. And I am from Ministry of Earth Sciences, and you can see we have two subordinate offices. One is IMD Delhi, which is looking after meteorological activities, and weather forecasting activities was looking after NCMRWF. And we have three attached offices. One of the office CMLRE, which I belongs to, and NCCR, which is dealing with the coastal research, and National Center for Seismology, and we have 5 autonomous bodies which is attached to the Ministry of Earth Sciences. And Ministry of Earth Sciences is serving to the country in the area of weather, climate, ocean, coastal change, hydrology, seismology, natural hazards, and explore marine living, non-living resources, etc., etc. And when it comes to CMLRE, we do research, we conduct exploratory surveys, we document the biodiversity of living resources, and this is the only nodal institute in the country which is dealing with the marine living resources behind 200 meters of depth. And we cover up to Indian EEZ. And we do work with the laws, regulatory measures, and policies when it comes to protection and conservation of marine living resources along the Indian EEZ, and which is located in Kochi. And the only center currently we have, it is CMLRE in Kochi. And this is the key areas, the broad areas that what we do in CMLRE. We do conduct ecological studies, biogeochemistries, maximum minimum zones identification for potential fishing zone activities, and biodiversity of entire marine living resources within the Indian EEZ. And we do deep sea fishery survey and some of the living enhancement program through marine ornamental fisheries, aquaculture. And it's mandate to introduce our ship, FORV Sagar Sampada, Fisheries and Oceanographic Reserve Vessel. And this is the only dedicated vessel in the country still today, which is dealing exclusively with the marine deep sea resources. And this is the base for our institute to establish in the memory of FORV Sagar Sampada, the CMLRE has arisen in 1998, and the Sagar Sampada is serving to the nation since 1984, and it's almost 40 plus years by today, and still it is contributing to the Indian marine living resources. And now get back to the topic. So advancing sustainable protein, where we culture the meat in the lab. When we talk about the cultured meat or smart protein or laboratory meat, so-called so far, some of the basic questions which arise to the normal people. And you might have heard this word somewhere in social media, somewhere in the Instagram universities, WhatsApp universities, etc. The first question is it really happening in the global sector? And second question, even if it's happening, whether it is safe to consume, whether it is suggested to consume for human consumption. And third thing is, most importantly, even if it is suggested to consume, whether it will come to our plate, means as part of middle class people in the India, whether middle class people can able to access this in the future. So before getting to that, let us look about the fish in the global scenario. So far, we all heard from last 2 days, aquaculture is the fastest growing food sector in the world, and India is stand tall. It is stand top second in the aquaculture production in the global contribution. And only thing we need to look at is the marine sector. And marine sector, currently we are producing around 17.5 metric million tons in India, including both inland and marine fishery sector. So when I look at the aquaculture sector, it is growing tremendously from last 2 decades. But when it comes to capture fisheries, where we all talking about the sustainability is very simple. We need to conserve something for tomorrow, that's all. We need to take care of the resource which we have, and we need to conserve the resources for tomorrow. And when we look at the production from the capture fisheries, majorly from the marine sector, the production state has remained stagnant from last 10 years. And even if you look at the stats of 2010, we are used to produce close to 4 million metric tons, and still today we are contributing 4.3 around. And this production is remained stagnant from last 10 to 15 years. That is where we need to look into, where we talk about sustainability. And why we need to do sustainability? Because the population is growing and the demand is increasing. And when we look at the per capita fish consumption, it has risen from 9 kg to 20 kg in 2018, and it is still growing on. And on the other side, when we look at the fish in the international trades, it has reached to 168, close to it is the data of 2024. 168 billion market share we have. And fish alone is contributing to the 17% to the total animal meat. And we can't compare the fish with any other meat sector. Why? Because of we have something unique in fish. We have this omega-3 fatty acids, which we can get it only from the fish. So that's why the whole world is somewhere we are looking into the nutrition, importance of the fish, and the world needs lands where the seas meet into the food sector. And when we look at the world food statistics, what we want actually, so how much food we require, whether we are getting the enough proteins to the public. So when we look at the data that says 1 in 7 people in Hungary, which is insecurity in the food sector. And 1/3 is we are doing food waste. And as per the agenda of 2030, we need to end up with a zero hunger. That was the target for us. And when we look at the global production, how fishery sector is contributing to the global sector. And when we look at the per capita in some of the small inlanding countries, which has increased to 50 kg, and as compared to 20 kg, what we are presuming globally. Due to all this, we required something to complement to the existing conventional method what we call aquaculture, what we call capture fisheries, and we need something more. For that, when we look at the Indian stats, more than 81% of the Indians eat non-veg, right? And I don't need to give an explanation because yesterday itself you can see the queue in our veg and non-veg counters. So no more, we may not require explanation why the people are turning towards the non-vegetarian because of the protein source and various other factors. And at the same time, when we look at the conventional meat production, we have some of the limitations, and to meet the global requirements and to meet the export opportunities, there is something more what we feel is required to feed the world. With all, and another important aspects, as we all heard, like we are the emission of the greenhouse gases, where we talk about the carbon footprinting. It's not only because of what we are doing with the fossil fuel combustion, it's also generating to the agriculture. Almost 24% of the greenhouse gas emission is coming from the agriculture sector, where we use the fertilizers and all these things. And among that, of course, 30% from the deforestation, we are destroying the, indirectly we are destroying the ecosystem. And 70% has been producing from the livestock production. And with, once we look of all other advantage, other limitation, like we use antibiotics in the poultry sectors, and just to obtain the growth immediately, you know, farmers are not waiting for the 2-3 years to get their profit. They want immediately, right? So for that, we use antibiotics to avoid from any diseases, and again it will affect us. And we all heard zoonotic diseases from last 10 decades. So we already suffered with lot of other infections like avian influenza virus and some influenza virus, especially in the poultry sector, which is very common. Still today we are suffering. And when we talk about the cultivated meat, I always talk about the strength of cultivated meat production. So when we look at that, the standard sustainable development goals, as per that, this cultivated meat technology will meet to address 8 UN sustainable goals. First is zero hunger, where we can complement to the existing conventional sector, and good health and well-being here, because we can control the requirements to the people. And innovations and infrastructure, and responsible consumption and production, and importantly climate change. We can address the issues which is related to the climate change, and SDG 14 is where we talk about life under the water, and of course life on land we can contribute. And overall, why we need all this? Because the One Health, where we need to take care of human health and animal health as well as the environment health. So this is where this cellular aquaculture concept has been arised. So, you know, this was not arised overnight. You might be hearing this word from last 2-3 years, probably maybe last 4-5 years. So, but it was initiated in 2001. It was basically initiated by the NASA, specifically for the space travelers, those who are staying in the space for the long time, and where they used to produce the meat from the goldfish for the space travelers in those days. So this has become a trendy from last 4-5 years, and people have started working on all these aspects. And meat, and he is the man, Professor Mark Post. He was the first developed the lab-grown meat in 2015, and he proved that meat can be generated from the lab. So after the Professor Mark Post has been initiated in 2015, this has become a trendy and globally, and all researchers are looking after, and all industrial sector is looking after this research. So what is, when we talk about the seafood production, what is basically required first? When we look at the meat, what it contains basically? It contains cells, right? And that is where the cells, when we talk about the fish cells, and so far, you know, cell lines has been utilized for various applications, started from virological studies. I'm not getting to each and everything. And recombinant protein production, biobanking and conservation, and genetic studies and reproductive biotechnology, etc. But we'll talk about the cellular aquaculture where we can use the cell line. And we may also know that in humans, we cannot use humans as a model organism. We use cell lines for the model as an in vitro tool for various drug toxicity and drug screening and toxicity studies. And, you know, the meat not only contains the cells, it also contains some of other biomaterials we call as, where we called as extracellular matrix, which is present in and around the cells. So what are, that also we need to complement, right? When we are talking about the meat, when we are going to produce the meat in the lab, we are not only producing the cells, we also take care of some of the biomaterial which is used to produce the meats. And that contains like various, like the extracellular matrix is composed of gelatin, fibronectin, collagen, etc., etc. Here you may have some doubts like, okay, the collagen I can also get it from seaweeds, I can also get it from plant source. Why can't we look after these animal sources? Yes, it is correct. You can also get the biomaterial from the plant source, and there are a lot of industries are working on plant-based meat, animal meat, to complement, to reduce the use of animal sources. And only thing we need to take care of biocompatibility with the animal cells, whether it is maintaining the muscularity and vasculation of the muscles tissue. So you can use anything. So collagen from any source, alginate from any source. So how it actually works? It works basically two ways. One is bioinks, what we call. Bioinks is contains the live cells and the biomaterials, what we are just I spoke about. Once you formulate the bioinks, which is called, we'll go for printing. You might have heard of like therapeutic application where the people are printing skin for humans, I'm talking about. They print ears and they do transplant, and they got success in many of the cases, even in India the people are doing. But whether it is economically viable, that's what my question. So we print it, we do incubation, and we'll keep it for maturation, and it will be remain in live condition. This is one way of doing the cultural meat. And second way, what we do is, we will extract the muscle cells, and we'll identify the right source of the cells, and we will culture in the large bioreactors. And once we complete with the mass scale production, we will do with scaffolds, what we called as biomaterials. So we will culture mimic the nature of naturality of the cells, and we will culture those cells with the scaffolds, and we will keep it for maturation, and it will attain the structure. The structure can be designed based on the scaffolds that we are going to design. This is another way of doing the meat production. And when we extract the cells, cultivate it, bioprocess, and this is the current status where we are now in India. And some countries are ahead of us, like Singapore and all, but we are still in the pilot scale, and some industries are producing. But when we look at the cost benefit, and somewhere we are working with the standardization of the mass scale production and for cultivation of the meat. And for meat means it's muscle cell, right? When we talk about the fish cells, there are only few reports are available still date about the fish muscle cells, and most of these cells has been utilized for various other applications, as I mentioned earlier, like virological, toxicological, etc. Only few cells has been nowadays that has been utilized for the cultivated meat production. So when I spoke about whether it is economically viable, once the Professor Mark post has started in 2015, 2016 itself, the industry has came up. This is called as Mampis meat, and they produce the meat, but which cost around $40,000 US to produce 1 kilo of the meat. So, which is behind our imagination and behind our affordability. And within the span of 1 year, when we look at the same Mampis meat in 2017, the cost has been come down almost 10 times down, which has come down to $2,400. And you can think of the only technology matters for today. And 2016, you can look at the industries, only 4 industries has been started working. Within span of, I am just mentioning few of them, now we have more than 150 plus industries which is working on. Within India, we have more than 30 plus startups and industries has been working in the area of cultivated meat. So let us look at the stats now. And whether it is available in the market, that's what many of them you may think. Yes, it is available, which is already approved by the FDA, and which is serving both in the supermarket and as well as in the restaurants. In the Singapore, which is good meat, which is a part of Middle East, which is previously known as Eats, it's available. But it's available, they are producing the lamb-grown chicken, and which is called as Mrcvbr, it is their restaurant's name. And real world's cost, when we talk about the current estimates for the retail production itself, for the current value, for today's value, it's cost approximately $100 per kg, which is still expensive, right? Which is coming around 8,000 rupees per kilo in Indian market, and for wholesale market, it's coming 5,000 rupees per kilo. But it's still, when you think about the middle class and common man, which is still above our limitation. But as per the prediction, the projected cultivated value to produce the 1 kilo of meat will come down to $5 to $6. It's coming around 400 to 500 rupees. That's what we are paying today, right? When you go for the chicken, hardly 150 to 250 rupees per kilo. But the technology is common for mutton also, right? Mutton, we are paying how much? We are paying close to 1,000 rupees for today. And when we look about the salmons and some of the marine fishes, which is so expensive, we are paying more than 3,000, 4,000 rupees per day, even today for the cultivated meat. So, the take-home message is that today is the technology that matters, and probably by another 10 years, we may get even in the Indian supermarkets as well. And these are all some of the industries that has initiated, and those who are working with the specific about some of the finless fish, they are working with the bluefin tuna, and Mampes meat, they are working with the salmon, and Siong meat, they are working with some of the crustaceans. They also launched the crab-based meat in 2016. They are expected to launch in this year. And these are all some of the companies which are exclusively working with the aquaculture species. Like when we look at all of them from marine sector, all of them, most of them cannot be cultured except some salmon and trout. And these are all some of, against some of the countries and some of the companies. So when we talk about when we need to attract the people to come towards the meat sector, right? Then when the most of the vegetarians, they won't eat meat because of various reasons, some ethical and other reasons. So for that reason, some of the companies which is exclusively targeting even the vegans, so they are producing culturing those cells, that extracellular matrix exclusively from the plant source. And even from marine source, some seaweeds and some tomatoes also, they are using some of the components from the tomatoes for designing the biomaterials for the cultured meat. And I would like to present some of our small work which we have done from last, we initiated in 2019 and we completed in 2023. And we choose Labio roita, and this is the species. Why? Because it is highly demanding and most cultivated species in India. And this species alone contributed around 5.1% to the global production. And it is naturally occurring in Indian waters. And those who are from Bengal, Odisha and northern part of India, you may have heard. Even southern part, Andhra coast and all, the people are doing this. And when we started working on this cultivated meat, first thing we all thought like, how we can, all aquaculturist question is same. How can we increase the growth? Everybody wants growth immediately, right? So when we look at the process of muscle formation in the fish, so there are several factors, several regulatory factors, several proteins that induces the growth in the fish, right? And one of the factor we all know that the whole world has already established is myostatin. This is the myostatin is the regulatory factors. What it does is, it will regulate the cell cycle in the G1 phase itself. So, that leads to the production of that P21 molecule that leads to the cell senescence, means it will lead to the cell death. So that automatically the growth of the fish will reduce. Now whether if we knock out this, whether we can able to achieve the maximum growth, that is the second question. And there are a lot of studies are going on, and it has been proven also, silencing and knocking out of some of the genes which is regulating the myogenesis process, and one of them is myostatin, which is also called as growth and differentiation factor 8. And once we knock out these genes, we can achieve at least 30 to 40% of the growth, which is already proven. And other than this, the muscle cells, when we talk about, this is not only like it's a process. It's a myogenesis process which is highly conserved and which is regulated by various myogenic regulatory factors. That's what we studied, and we have undertaken from the satellite cells to until the formation of muscle fibers, how the cells differentiate themselves across the each stages, and which stage of the cells which is more suitable for the cultivated meat. And we didn't end up only with the regulatory factors, we also considered some of the proteins which is crossing simultaneously against each regulatory factors, and we have studied the whole proteome expression across the myogenesis process, and then we came to the conclusion, yes, there is a similarity between the regulatory factors like genes and some of the proteins which is expressing simultaneously. And we have selected some of the genes which is already known, and we perform the whole proteome by using LCMS Orbitrap, and we have used some of the softwares for data analysis, and we do the primary culture, and we standardize the growth parameters and some basic authentication for the cells we have done. And these are all the various myogenic regulatory factors that has expressed across the stages. What we did is we have induced the differentiation within the cells by using various growth factors, and we studied the complete myogenesis within the muscle cells. And we mapped the whole proteins. What we obtained, we found more than 15 unique proteins across the myogenesis process, and we mapped them, everything, and the Venn diagram, when we look at the Cs and their abundance and their functions across the each stages, what's the regulation, what's the role of each proteins across the each stages of the cells, and even the protein-protein interaction also we have studied, and what is the role in the metabolic activities and carbon metabolism, etc., and ribosomal proteins. We identified the proteins which is responsible for the production of and each across the each differentiation of the cells. And here some of the unique things we found. We even went ahead and we identified which are the proteins within that fish which we choose, which is responsive of bitter. And because we may all know that we have our own reasons to not to eat something, right? Some people will not prefer freshwater fish because of presence of intramuscular bones. They will say, I don't eat because of bones. Some people say because of like high amount of fat, we will not consume it. But here everything is under control, and we identified each protein which is responsible for salt, sour, sweet, bitter, umami, etc. And we cultured with the scaffold, which is basically made from mushroom. We extract the component from the mushroom, the alginate, collagenate, whatever from the mushroom. With that, we designed the scaffold, and we do collaborated with the startup and with the IIT Bombay. And we cultured ourselves inside the scaffolds, and we achieved. And finally, we transferred the technology to the startup, and they are taking ahead of all this, what we have done preliminarily. Now, the need of an hour, and I hope I answered all the questions which I asked myself in the beginning. Whether it's happening, yes. Whether it is safe, yes, it is safe. It's already proven. It is approved by the FDA, and it's already in the restaurant. And economically, we may think like, yes, it will happen. Probably it may take another 10 years of time. Probably you all will remember, there was a guy in the conference at Vivekanandha College who presented the cultivated meat. After 10 years, you may look into our restaurants as well. But what is required for today we need to work on the development of appropriate muscle cells. We need national and international collaborations, mass scale production, and by identification of novel bioactive compounds which is required for the to mimic the natural mechanism in the cells. And finally, the culinary effects. What is meant by culinary effect here? Like we need to get the same taste as we are getting in the natural conventional method, right? Yesterday we all eat chicken. That what we grown in the lab also should give the same effect. And while cooking as well as while consuming. That is finally left to the consumer. We also taking to part of that. And future prospect, when we look at the convention, when we compare this with the conventional methods, which is having its own benefits. When we talk about the animal welfare, the single sector cannot contribute to the fullest to meet the requirement of the protein to the country. And of course, the quick production. When we talk about the production, when we go with aquaculture, we need to wait for minimum of 1 year, right? To attain the marketable size of the fish for approximately 1 to 1.5 kilo. But here we need not wait. And functional designer meat, as I already said, this we can control everything within the lab. How much fat required, how much protein required, based on the consumer preference, we can work on the entire nutrition is under our control, because nowadays we are looking after more into health aspects. And of course, the decrease in meat contaminants and meat-borne infection, we usually getting from the poultry sector and even some of them from even aquaculture sector. This all can be ruled out. And this is what stats says. After the effect of COVID-19, we all of a sudden will become a health conscious. And the study has been conducted by the US. Why you people need to consume the seafood? And based on the response from the consumers, 2/3 of the seafood consumers says they are moving towards health and nutrition. That is the primary and foremost rule for the life. And 74% of the consumers, they still wanted to know the more beneficial about the nutritional factors from the seafood. And still 71% of the seafood consumers want to know more about the seafood sustainability. Because as I already said in the initial presentation, our marine sector, the contribution from the marine sector will still remain constant from last 10 to 15 years. So where the sustainability matters for the future. And with all, this is the data 5 years back, where the lamb-grown meat gets the rare funding boost. And still today, there is a lot of funding agencies, even in India and globally, they are supporting in huge, and even some of the India has haven't take up, and Maharashtra government also, it is plans to, now it's already there in the cell-based research, and they're supporting a lot of meat startups which is working on the culture-based meat production. With all, I would say the future is near before we think, right? And I would like to thank all the organizers, and especially to invite me, and it's my pleasure to be here today, and I would like to thank all the organizers of this conference for making me time for today's presentation. Thank you one and all.
We thank Dr. Yashwant sir for the insightful keynote address and valuable scientific inputs. Thank you, sir.
Thank you everyone.
Thank you, Dr. K. Anandaraja sir, for your kind information, which is very useful and very inspirational. I hope all participants give information from sir. So thank you very much, sir. So next I would like to invite Dr. Kandaraja sir, scientist, scientist from Germplasm Exploration, Evaluation and Conservation Division, ICAR National Bureau of Fish Genetic Resources, Ministry of Agriculture and Farmers Welfare, Government of India. He is working as scientist in ICAR NBFGR, and he published more than 180 papers in reputed national and international journals. He also published more than 16 book chapters, and he also worked as the co-invigilation for many R&D programs, and he also worked as the co-invigilator for the many major and minor funded research projects. He also worked as the organizing secretary and convener, and also worked as the coordinator for many extension and capacity building programs. And he also published very popular articles in Nature and SCA journals. And he is the published many manuals recording the biodiversity of the fish genetic resources. And he also discovered new species in new 4 species. So he also completed many short courses, summer courses and refresher courses. So in this regard, I would like to Dr. Gandharajan sir. So welcome you sir. And now the session is hand over to Dr. Kandarajan sir. So all the participants are requested to utilize this section.
Thank you, ma'am. Yeah. Thank you, madam. Very good morning to all. Now I am going to present the topic like fish genetic resource conservation towards sustainable blue economy. So from yesterday we are talking about the marine ecosystem and its conservation. How are we going to utilize this ecosystem in a sustainable manner? So many questions are coming. So what is the solution? How are we going to use this ecosystem in a sustainable manner? There are so many challenges are coming in front of us. So many threats are there. And in the future, how we are going to use this ecosystem in a sustainable way. So I will brief some of our activities, ongoing activities and completed activities, how we address these challenges, and in the future, how we are going to maintain the sustainability in the ecosystem. So I am Dr. G. Kandarajan, working as a scientist in the Center for Peninsular Aquatic Genetic Resources. It's a regional center of ICAR, National Bureau of Fish Genetic Resources. And this is a brief introduction about the institute which I belong, ICAR, National Bureau of Fish Genetic Resources, established in the year 1983. Our headquarters, institute headquarters is located at Lucknow, Uttar Pradesh. And we are mandated to explore, characterize and catalog the fish genetic resources of our country, including freshwater and brackish water resources, marine resources. And we are mandated to maintain, at the same time preserve the fish genetic resources, not only for the conservation, but also for the utilization of the resources. And we are also mandated to evaluate the indigenous and exotic germplasm, including their risk assessment and fish health. So the administrative wise, we have our institute has 3 divisions at the headquarters. Genomics and Computational Resources Division, Germplasm Exploration, Evaluation and Conservation Division, and Exotics and Aquatic Animal Health Division. And we have a regional center, that is Center for Peninsular Aquatic Genetic Resources, to look after the peninsular fish genetic resources of the country. And we have one training unit located in the outskirts of Lucknow city. And yes, when we talk about the genetic resources of our country, if we see like in the world where we are standing, so India is one of the 17 mega biodiversity country in the world, and we have 7 to 8% of the recorded species of the world. It is present in India, and more than 54,000 recorded plant species are there in India, and we have more than 1 lakh recorded species of animal diversity in the country, and we have 4 biodiversity hotspots. In the world, there are 34 global biodiversity hotspots, and of which we have 4 biodiversity hotspots, that is Western Ghats and northeastern part of our country, Himalayas, and the Andaman Nicobar Island. And when we talk about the aquatic genetic resource of our country, aquatic genetic resource, it includes the finfish and invertebrate genetic resources, which is having the potential value, contribute to the production. And globally, there are 36,000, more than 36,000 fish species are reported, of which roughly 9% of the global fish diversity is present in India. So we can see the fish genetic potential of our country. And when we talk about the finfish species, we have more than 3,000 fish species in the country, and of which more than 950 fishes are present only in the freshwater ecosystem, and rest are the marine fish species. And molluscan species, more than 3,200, crustacean, around 3,000 species, echinoderms, more than 750 echinoderms, hard corals, around 218 hard corals. So we can see the rich fish diversity of the country. When we talk about the fishery resources, it includes all our water bodies. We have the coastal length of 11,099 kilometer coastal length, and exclusive economic zone of 2.02 million square kilometer, continental shelf, number of fish landing centers located along the coastal area of our country, and rivers and reservoirs running more than 2.5 lakh kilometer in length, and reservoirs more than 4.03 million hectare, tanks, ponds, beels, oxbow lakes, freshwater resources. So in terms of genetic resources as well as the fishery resources, our country is very rich. And the status of fish production, we can see the total fish production is 19.5 million tons. We are the second largest fish producing country in the world, and the fishery sector is contributing 1.09% of the gross value added product of our country. And we can see our sector has grown from the capture fisheries to culture fisheries. In the 1950s, we were mainly capturing the fishery resources from the marine systems, and the inland sector, that is mainly aquaculture sector, was contributing very less. Now in 2024-25, the trend is almost reversed. So now main production is coming from the inland sector, and the fish production from marine sector has slowly diminished, and now it is in the stagnating phase. So we can see the changes that had happened after independence in our country. So fish genetic resources we are talking. This fish genetic resources, it is linked to all sustainable development goals. You take sustainable goal number 1, no poverty, to sustainable goal number 14, life below water, and you take the sustainable goal number 17, partnership for the goals. So fish genetic resources are linked to all sustainable goals of the United States. And fish genetic resources is the main agenda for achieving the blue economy. So we have the vast fish genetic and fishery resources, and there are millions of fisher folk depends on these resources directly and indirectly. We have fishermen going for fishing, directly contributing, and we have the fish processing plant, we have the marketing sector. So there are crores of livelihood depend on this genetic resource. So with vast maritime resources, inland freshwater resources, we have the scope to accelerate the production of our country, contributing to the blue economy. And this particular blue economy, it is directly linked to the SDG 14, that is conserve and sustainably use the oceans, seas, marine resource for the sustainable development. So what is the relevance of fish genetic resource conservation? We have the agriculture sector, poultry sector, dairy sector, but there and all we are not talking about the resource conservation. Only in the fisheries we are talking about the fish genetic resource conservation. Why? Because the animal farming and agriculture sector, it has the, means production is coming from the domesticated varieties. So there we are not talking about the conservation of the wild resource. Rice it is being grown by us since long back. But in the case of fisheries sector, as we have seen in the earlier slide, in 1950 we were mainly depends on the capture fishery resources. Now only we are moving towards the culture fishery resources. So the fish genetic resource conservation is still relevant because most of the fishery resources are still it is coming from the wild-based resource. So that's why we are talking about the fish genetic resource conservation. Then what are the threats? We have so much resources, but at the same time these resources are highly vulnerable to various threats, like habitats are being destroyed due to the hydrological alteration, dam construction, e-flow modification, mining, and when we take aquatic pollution due to the heavy metal poisoning and pesticide poisoning, eutrophication issue, the habitat quality is being degrading day by day, and we are exploiting the resources from above the optimal limit, and the invasive alien species. The indigenous fish species are threatened by invasive fishery resources. So these are all some of the threats. Apart from that, on top of that, climate change is influencing all this threat, and it is influencing the fish genetic resources. So climate change, when we take seriously, it is affecting the coastal ecosystem by coastal erosion, sea level rise is happening, flow modification is there, then fish physiology and health is changing, exotic is spreading very fast and it is affecting the indigenous species. There are so many extreme weather events are happening, cyclones are happening, heat waves are happening in the inland region, and eutrophication, global warming, ocean acidification. There are so many threats associated with the aquatic ecosystem because of this climate change. So now our resources are there and it is highly threatened due to the man-made and climate change activities. So we are bound to conserve our aquatic resource to maintain the sustainability. So there are various ex situ and in situ conservation methods in which the fish genetic resources are being conserved. We have the protected areas, fish sanctuaries announced in various parts of the country. It is conserving the wild fish genetic resources. We are studying the life history traits of natural fish species, where it goes, how it migrates, where it breeds, what is the fecundity, at what time it breeds. So we are monitoring, we are documenting the life history traits. We are continuously exploring the resource. There are so many unexplored resources are there in the country. We are continuously doing exploration. We are doing captive propagation. After studying the life history traits, we are collecting the fish species from its wild habitat and we are studying its habitat requirement, and we are developing the breeding technology. There are other concepts are there which we will see in the upcoming slides. And these are also marine protected areas announced by the government of India to protect the wild fish genetic resources. Here we can see the Gulf of Kutch, Gulf of Mannar and Sundarban area, and there are so many national parks in the Andaman Nicobar region. And apart from that, we are so many sanctuaries, Goringa Sanctuary, Silka Lake, Krishna Mangrove Sanctuary and Pulikat Lake and Bakpe region for sea cow. We have so many protected area announced to conserve our marine fish genetic resources. And from our institute, we are conducting regular exploratory survey. You can see from the year 2017, we have covered so many riverine ecosystems, so many wetlands we have covered. If we talk about the coastal region, we have covered from the Gujarat to West Bengal, and the island resources, Lakshadweep Island, Andaman Nicobar Island. We have covered the entire part of the country to document the aquatic genetic resources. And if we take the riverine ecosystem, we have covered the Luni River, Banas River in the Ganga Basin, and Mahanadi River, and Godavari Riverine ecosystem, Kaveri River, and various Ramsar wetlands of the country. We have covered diverse ecosystem to document the aquatic resources. And through this exploratory survey, in the course of time, our institute has discovered more than 60 new finfish species, and 8 shrimps, and 1 cone snail, and 2 fish parasite, and 10 we have rediscovered more than 10 fish species. So we have created the DNA barcode record for more than 600 fish species, and we have documented the genetic resource information. We have created 4 databases, and these are all some of the fish species discovered by our institute. And we have conducted exploratory survey, intensive exploratory survey in the Lakshadweep Island. You can see the colorful invertebrates, shrimps documented through the exploratory survey. And we have conducted more than 200 exploratory survey covering the 421 locations in the Lakshadweep Islands, which lead to the discovery of 4 shrimp species. And these species is being collected and it is being maintained in our live gene bank. And the local resources, because of the exploration, we are collecting the local resources, and these resources is being maintained in our local live gene bank in the live condition. You can see we have the live gene bank for Ganga River in the Lucknow, and for the northeast region we have the live gene bank in the Guwahati region, and we have the live gene bank for clownfish in the Maharashtra. And for Krishna River peninsular fish resources, we have live gene bank in Nagarjuna Sagar Reservoir, in the Lakshadweep, in the Cochin for Western Ghats fishes, and in the Pichavaram for Gulf of Mannar region fish species. So these live gene banks are located in various agroclimatic zones of our country, and documenting the endemic resources. And this is the view of live gene bank located in the Lakshadweep Island. Here you can see the facility and the shrimp species being maintained in the resources. As I said already, we are not only documenting the resources, collecting the resources, studying its requirement, and we are developing the breeding protocol for its young generation. You can see the hatchery facility of our Lakshadweep facility. And these are all the beautiful shrimp species which are collected through our exploratory survey, and we are maintaining these species in our live gene bank located in the Lakshadweep to target the blue economy through the ornamental fish culture. And cryopreservation is another promising technology for the fish genetic resource conservation. We all know, like, cryopreservation means conserving any biological material using liquid nitrogen at minus 196 degrees Celsius. So through this technology, what we are doing, we are collecting the eggs and embryos from the endangered and endemic fishery resources, and we are doing cryopreservation. We are maintaining in our cryopreservation facility. And for your information, we have the cryopreserved milt of our Indian major calves. It has already crossed more than 25 years. So more than 25 years that we are maintaining the genetic viability of the organism, and we have revived, and it has given the promising revivable capacity. So this is one of the promising technology, and in our institute, NBFGR, we have developed the cryopreservation protocol for more than 35 fish species. And we are collecting the individual, we are maintaining in our live gene bank, we are documenting its life history traits, and we are developing the breeding technology. So after the breeding technology, what we are doing is we are developing the young one, and we are ranching the young one in its native ecosystem to build the natural stock. So here you can see such activities are continuously taken by our institute through the Pradhan Mantri Matsya Sambada Yojana in 3 river basin, Ganga, Brahmaputra and Barak, Mahanadi river basin from October 2021. So initially under this program, this river basin is covered, and from our NBFGR, we are doing ranching program in these 3 river basin, and apart from that, for endemic fishes, we are doing regular ranching in the Western Ghats also. So apart from NBFGR, other institute like CMFRI, They are doing ranching program in the Gulf of Mannar and Parkway region. They are regularly ranching the post larvae of shrimp species in the natural environment, and other fisheries institute also, they are doing regular fish ranching in the natural ecosystem. So whatever information we collect through the exploratory survey and documentation, we are documenting in the repositories, and we have developed the repository called Aquatic Genetic Resource Information System of India. So you can visit in this website. It is having the information of all fish species reported in our country, and you will find the information of its distribution, biological information, all related information you can find from this website. And apart from that, genomic information is being maintained in the portal called Fishomics. You will be able to get the barcode information of the fishes, mitochondrial DNA information, all genomic related information you can get from this portal. And there is a concept called state fish. We have our national animal, national bird. So likewise, we have the concept called state fish. So each state, this is the concept proposed by our NBFGR. And in the concept, in this initiative, each state encouraged to identify one fish. And most of the states, around 22 states, they have announced, they have identified one state, which is culturally important, local preference, ecologically important to the particular state, and it is contributing to the historical significance of the country. So around 22 states, they have identified their state fish, and you can see from Jammu Kashmir to Kerala and Arunachal Pradesh to Gujarat, they have identified around 17 fish species, and the conservation initiatives are being continuously taken by these states for the promotion of aquaculture and conservation. And there is a concept of conservation aquaculture. Aquaculture, we know, like culturing any animal in the controlled condition. So conservation aquaculture is, this is the use of aquaculture for conservation and recovery of endangered species. So here we cultivate the endangered species and endemic fishes. In the process of aquaculture, we are also contributing to the conservation of these resources. And there is a successful model implemented by our institute, that is community aquaculture for the livelihood development and the conservation. So this is the initiative being implemented by our institute in the Lakshadweep Island. So here, as I have already informed, we are doing exploratory survey. During the exploratory survey, we are collecting the individuals. We are maintaining these individuals in the hatchery. And after the developing the breeding technology, these young ones are being supplied to the local beneficiaries, mainly the islanders. They are growing, they are doing this activity as a backyard activity, and they are cultivating these shrimps for 2 to 3 months, and after that, these shrimps are being sent to the export and domestic aquaculture, domestic ornamental sector. So through this way, it is contributing to the livelihood promotion of the islanders. So you can see the nano aquaria, and we have tied up with aquaculture industries in the country. So they are regularly collecting the individual from the farming community. So they are earning regular income through this approach. So you can see these are all the clownfish species and shrimp species for which breeding technology is already developed, and the local community is taking up this activity for their livelihood promotion. And these are the community aquaculture units established in the Lakshadweep Island, and 46 women. We have trained more than 80 islanders, and of which 46 women, they are regularly involved in the culture of this shrimp. And this is their culture unit, and we can see the smiling faces, which are they are earning more than 7,000 through this activity. And as an additional income, they are doing their regular work, and they are spending 2 hours or 1 hour daily to maintain this shrimp species. Then after growing more than 2 to 3 months, they are selling this and they are earning the profit. So this model, this community aquaculture model was highly appreciated by the Honorable President of our country, Draupadi Murmu Madam, during her visit to our facility in the Lakshadweep Island. So this facility harmonizing the biodiversity, it is contributing to the conservation as well as for the livelihood promotion. It complies with the national and international goals, and this facility is environmental friendly. It is not producing any emission to the environment, and whatever organism we are promoting for aquaculture, these are all the indigenous organism. So we are not promoting the non-native species. Through this way, we are contributing to the indigenous conservation of indigenous organisms. So the same concept we have implemented in the mainland with the support of mangrove cell in the Maharashtra state. So here you can see this initiative is concentrating the promotion of indigenous clownfish species. We all know clownfish species. So we have developed the breeding technology for some of the indigenous clownfish species that after developing the young ones, these young ones are being supplied to the local fisherman community. They are growing this for 2 to 3 months in the backyard, and after that these well-grown young ones are being marketed through the aquaculture industry, and they are earning regular income. So we have set up the marine ornamental fish village in the coastal districts of Maharashtra, Ratnagiri, Sindhudurg region. So the fishermen of this region earning regular income through this initiative. And the same initiative we have taken up in the Pichavaram region of Tamil Nadu. So we have collected the indigenous clownfish from Kalpapannar region, and the same way we have developed the breeding technology and standardized the breeding technology. The young ones are being regularly generated, and it is being maintained, it is being supplied to the local tribal fishermen, and they are doing the cultivation, and it is supplied to the beneficiary. So we can see the hatchery facility which is located within the Faculty of Marine Science campus in the Annamalai University of Parangipettai. And the fish cell line, I no need to explain this because already previous speaker Yashwant has given a brief overview of cell line, what is cell line and what is its potential. So one thing I wanted to emphasize here is the NBFJR is the nodal agency established the National Repository of Fish Cell Lines at the headquarters Lucknow, and we have more than 87 fish cell lines of different fishes which is being maintained in the National Repository of Fish Cell Line. So this is the largest fish cell line facility in the world. So researchers, those who want fish cell line for their cytotoxicological study, they can contact the National Repository of Fish Cell Line. So they can access the fish cell line facility also. So this is another aspect of a flagship program of our institute. We are coordinating the fish disease surveillance. So aquaculture activity is very rapidly picking up in the country. So at the same time, we are facing the issue of diseases in the aquaculture system. So we have coordinated the network with all the state fisheries department, fisheries colleges, and other universities of the country. And the fish diseases reported by the aquaculture farmers are being regularly monitored, and we are providing the support for screening the diseases, and we are providing the measures to overcome these diseases through this national surveillance program for aquatic animal diseases. This program is being implemented through the Ministry of Fisheries and Animal Welfare. So this is the National Fish Museum and Repository located in the headquarters. So this is the recognized repository for transfer of fish resources by the Biological Diversity Act and NBA. So we have the, in the presentation I have shown, we have more than 3,000 fish species. So in this museum, we are maintaining the voucher specimen for more than 1,200 fish species reported from our country. So who are doing taxonomy related work, they can visit the museum facility, so they can compare the voucher specimen before describing any new species. And we are maintaining the more than 250 voucher molluscan species. We are maintaining more than 19,000 fish tissue accession, fish cell lines, fish melt, and we have the radiography facility, X-ray radiography facility for fish species. So these are all some of the flagship activity which our institute is taking to conserve the aquatic genetic resources. So when we see the way forward to which we can conserve the aquatic genetic resource, so the community participation is very much important to conserve the endemic and endangered fish species resource. For that, the participation of the fishermen community and other stakeholders are very much necessary. And the climate vulnerability assessment. So climate is affecting the aquatic ecosystem, fish species. So we have to assess the resilient capacity of the ecosystem and the fish species to overcome the climate change, means adverse impact of climate change in the future, and restructuring the existing legislation. So we have the National Sanctuary, Protected Area, we have the Wildlife Act, Environmental Protection Act, everything we have. But it is time to restructure the activity considering the threats faced by this aquatic organism. There are so many advanced tools. eDNA is there, microbiome approach is there, AI is there, drones, remote sensing, ecosystem services approach. So it is time to integrate all these approaches to achieve the sustainable utilization and conservation of these resources and the ecosystem service perspective. So when we talk about the aquatic ecosystem, we see the ecosystem only with respect to the fish production. Apart from fish production, this aquatic ecosystem also providing various other ecosystem. So we have to see the aquatic ecosystem in that angle. So to expand this horizon, we have to see the ecosystem through the ecosystem service perspective. So we have to take the monetary value for our conservation initiative to bolster the conservation activities, and we have the other unexplored ecosystem, deep sea ecosystem. So the exploratory activities are being regularly carried out by so many research institutions. So it is time to accelerate the activities in this unexplored ecosystem. And another major aspect is diversification of aquaculture. We have very handful species for the aquaculture diversification. So it is, but at the same time we have more than 3,200 fish species with us. So it is time to diversify the aquaculture sector of the country, considering the climate change and other issues we are facing. So this is all about from my side. So at the end, I would acknowledge our director, ICAR NBFGR, and Dr. T.T. Ajit Kumar, Principal Scientist and Head of our division, the man behind all the community aquaculture related activities in the Lakshadweep Island, and the funding agencies supporting our initiative, ICAR under Ministry of Agriculture and Farmers Welfare, DST, CMLRE under Ministry of Earth Science, and Mangrove Cell of Maharashtra state government. So thank you all for the opportunity.
Thank you, sir, for your thought-provoking and inspiring address.
Thank you very much for a wonderful presentation, sir. For delivered excellent speech on fish genetic resources conservation towards a sustainable blue economy. The topic is very clearly and effective for our participants, sir. Thank you so much. I want to say, thank you.
Have a break, have a tea. The refreshment has been arranged in the dining hall. We kindly request the participants from other colleges to proceed first and take your refreshments and be seated back in the auditorium within 10 minutes. We request our VR students to please wait for a while. Once again, I repeat, the refreshment has been arranged in the dining hall. We kindly request the participants from other college to proceed first and take your refreshments and be seated back in the auditorium within 10 minutes. We request our VR students to please wait for a while.
கோடிங்னாலே பயமா இருக்கு, ஆனா ஐடி ஃபீல்டுக்குள்ள ஒர்க் பண்ணணும்னு ஆசைப்படுறீங்க.
Data-driven decision making, policies succeeded not on paper but through.
Welcome our Director Paramedical, Captain S. Gokulanathan sir, to this occasion. I also extend a warm welcome to the fellow principals, Dr. S.K. Rajkumar, Dr. Sendil, Dr. Sesinda Madam, Dr. Sumadi Madam, and Dr. Harness Madam, and Vice Principals of both the colleges to this function. I also extremely happy to welcome all the parents. You believed us and
Inflation healthcare not only mere economic objective, this
Vivekanandha education.
During 2000,
to be part of this prestigious graduation day ceremony at Vivekanandha. They operate in the extreme environments like -50, +50, underwater, in the air, jungle. So it's huge task. So we develop the combat casualty care and trauma management in the extreme environment, including the forward area, including space, and biomedical instrumentation and diagnosis for chemical, biological, nuclear disaster. Protective healthcare system, including protective like antidotes, vaccines, protective suit, etc., and human performance and life sciences in the extreme environment. Many defense biomedical innovations find direct application.
welcome with applause.
And Professor Dinesh for their continuous
Students, please be seated. The session is starting now. I request all the students to take your seats. Students, please be seated. The session is starting now. May I request all the students to kindly settle down as the session is about to commence. Students, please be seated. The session is starting now. May I request all the students to kindly settle down as the session is about to commence. For oral presentation, the venue is room number 11 and 44. The venue has been shared in the WhatsApp group. Kindly check the details and proceed to the venue. All the participants are requested to kindly proceed to the oral and poster presentation venues as scheduled.
In the memory of FORV Sagar, any other meat sector.
We extend a warm and respectful welcome to our honorable guest, Dr. D. Dinesh Santa Lejoni Maurabain Sir, Principal Scientific Officer, Rajiv Gandhi Center of Aquaculture, Ministry of Commerce and Industry, Government of India. His expertise and contributions to aquaculture research add great value to our honorable guest, Dr. D. Dinesh Santa Lejoni Maurabain Sir, Principal Scientific Officer, Rajiv Gandhi Center of Aquaculture, Ministry of Commerce and Industry, Government of India. His expertise and contributions to aquaculture research add great value to our honorable
University and MSc in Marine Biology and Oceanography and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture research. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture research. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture research. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture biology. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture biology. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture biology. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture biology. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir has been a pioneer in marine fish and shrimp aquaculture biology. Sir has also undergone international training in biology and Indonesia and his BSc in Zoology. With over 20 years of experience as a Principal Scientific Officer at RGCA, along with 4 years of Senior Technical Assistant and 7 years in private aquaculture industry, Sir
Sister Society under MPDA. and it promotes diversified aquaculture by producing quality seeds for various commercially important export-oriented aquaculture species. RGC operates around 13 projects all over India across all the coastal states, and all the projects are planned scheme of Department of Commerce, Government of India. So all the projects are the first of its kind on a commercial mode. It's all not purely on research, it's fully on commercial scale. So this RGC is registered with Bharathidasan University, Tiruchi, and also supporting PG students for their internship. As a part of diversification of aquaculture, RGC is having about 13 projects all over coastal states. The Seabass Hatchery is producing commercial seed production at Toduvai. It started in 1999 and supplies seeds for all over India. Also, Mrcvbr, it's only one hatchery under government agency. It is located at Toduvai. It started in 2004. And aquaculture demonstration farm where it demonstrates crab culture as well as sea bass culture set in pond cages. Then domestication of tiger shrimp project. It is one of the national project in India, and Artemia project and demonstration farm working in Ramanathapuram, and RGC headquarters is situated at Sirkali, and grouper project is in Kodiaghat, Andaman, and aquatic quarantine facility for L. Venam is located at Nilangare, Chennai, and gift genetically improved farmed tilapia project in Manikonda village, Vijayawada, marine finfish project in Poriyur, then open sea cage project in Muttam, and L. Venami BMC project in Vizag, and multi-species aquaculture complex at Cochin. Now I am going to share some of my experience. I worked in a grouper project. Grouper, one of the high value food fish, it fetches high value when it is sold in live form. You see the grouper production continues to expand. It's about 10 to 77% per annum since 1999, and this fish is highly prestigious fish, and high demand in the South Asian countries, especially Hong Kong and China. And this estimated live fish market in Hong Kong, China is about 20,000 tons per annum. The seed production culture will reduce because this is most of the groupers are under endangered fishing pressure. So if you do the aquaculture production of this fish, it will be conserving the fish as well as and commercial production of aquaculture. So this is a live fish grouper fish market in Hong Kong and China. So this live fish will be displayed in the aquarium. The entrepreneurs or the buyers will come and select the fish, and they will check that fish and for food preparation. These are some of the 4 commercially important fish, especially for groupers. The first one is Ephinophilus phascocutatus, the tiger grouper. It fetches around $30 to $35. It is a medium priced fishes in the US market, in South Asian market. And in that one, another one is Chromoleptis altivelis, that is mouse grouper. It takes, it goes to market size about 1 year. It fetches around $60 to $75 in the Hong Kong live fish market. Then another one is Ephinophilus coides, orange spotted grouper. It fetches around $20 to $25 a kilo. And the Proctorpharmus areolatus and the squirrel tail coral grouper, $50 to $60. All these grouper varieties are readily available in Andaman, except Ephinophilus coides. All fishes can be, other 3 fishes can be grown only in cage culture, cage with full strength seawater. Whereas this Ephinophilus Ephinophilus coides can be grown in the brackish water. Now I am going to talk about the Ephinophilus breeding program of Ephinophilus phascocutatus. We developed the breeding technology, hatchery technology for the Ephinophilus phascocutatus. So we selected Andaman Islands for this project. Why? Because it has got excellent potential for grouper aquaculture in Andaman. There are about 35 grouper species available in Andaman, reported by P.T. Rajan. And among the 35 species, 12 species are very well amenable for aquaculture. And also good availability, good quality of broodstock, because we did a survey and found that most of the grouper species are free from VNL, virus nervous necrosis viruses. Also, this Andaman Islands is bestowed, naturally bestowed with a clean, pristine environment, and there is many protected areas where we can do the small-scale cage culture of this species. These are all the identified areas and earmarked by the Andaman administration, and recently they released in the Gazette notification to carry out cage culture of marine food fishes. This is Ephinophilus phascocutatus, commonly called tiger grouper. In Tamil it is called Kalavai mean. In Hindi it is called Pattar Gobra. It is a moderately priced fish, and the taxonomical position is given below. And this species we selected because it is available plenty in Andaman. And before starting the project, we should have a brood stock first. Without brood stock, we cannot do the hatchery seed production. So for getting the brood stock, we have to condition the fish in a cage in a natural environment. For that, we did a survey and selected a place to put up the cage. So these are all the criteria we need to select site selection. First, you should have a clean water away from pollution and other things. And water depth should be minimum 15 to 20 meter depth water should be there, because the cage depth will be 3 meters below which, and there should be a free flow of water. Underwater current should be 10 to 30 centimeter per second. If it is more than that, your cage structure will be washed away by the underwater current. Then safety in the sense should be away from the poaching and other areas, other things. And the temperature should be minimum 28 to 30 degrees. Then only the growth of the fish will be good. Then anchoring possibilities. If the area is too much depth, anchoring will be very difficult. And we cannot do anchoring and taking, and difficult anchoring will not be possible. So at least 15 to 20 meter depth and a suitable bottom without rocky area should be there for anchoring. Then proximity to infrastructure. The cage structure should be very close to the shore, so that we can easily go to the shore and come back for transporting fresh water, feed, and personals. And very importantly, it should be away from wildlife sanctuaries. Why? Because it is wildlife sanctuaries, Andaman Nicobar Islands, most of the areas are covered under wildlife sanctuary. So we have to be very careful to select the place to put up the cages. So we selected this place called Rutland Island, which is about 3 kilometers away from Chiriatapu. We have to travel by boat by 3 kilometers by open sea and to reach our cage site. This is a semi-protected area wherein we put up the cages. This is the cage structure we put up in Andaman Islands. Initially we started with wooden cages and started collecting the brood stock. And in due course of time, after 2-3 years, the wooden cages is not withstanding because of and what we did, we planned to change to HDP cage. Very wooden cages. This is the HDP cages. This is the base frame, and the cage HDP pipes are welded together with the butt welding system. After butt welding and making a base frames, this will be towed by sea and reached to the cage site. HDP cages, the bottom 50 mm dia pipe was used as a floating pipe. Pipes together. This is the basic by using HDP pipes. All these HDP pipe materials and other accessories are readily available in India. This is our fully made with the HDP pipes. We have around 21 HDP cages in the open sea. In that, we 7 cage rafts exclusively for maintaining the brood stock, grouper brood stock, especially the tiger grouper. The other 14 cage rafts are used for a cage farming of tiger grouper. And 2 platforms, 1 and 2, two is for treating the fish, another 1 is for keeping the cage materials such as anchors, nets and other things. And number 3 is people used to stay there for 24/7. Otherwise, sometimes poachings will happen, and there should be some personnel should stay for 24/7, even during hard times also. So these are all the anchors, different type of anchors we used for mooring the cages. That is Samson type anchors, the grapnel type anchors, and this one plough type. This plough type anchor is about 400 kilo weight, and Samson type anchor is 50 kilo weight, and grapnel anchor is 30 kilo. Depend upon the area we use this type of anchors. Also, these are the net cages. The groupers are having very soft skin, very tenoid scale. So we should have a double layered net cages. This one is black color, one is nylon knotless net. You can see there is no knot, and the other one is HDP knotted net. So these are a double layer net. Inside it is nylon knotless net. Outside it is a nylon knotted net. To protect nylon knotted, HDP knotted net is used as a protecting net, and nylon knotless net is used for the holding the fishes. These are the different types of fish cage size: 12 mm, 16 mm, 25 mm. 12 mm is used for the earlier stage of fingerlings, and 16 mm for the medium size or sub-adult stage, and 25 mm is for the broodstock size. So how you collect the fish and quarantine? Mostly the craft and gear we use for for collecting fish is the traditional dongi we call as dongi or boat. Dinghy is called in Hindi. It's called boat. We used that boat to collect the fish by using hook and line method. After collecting the fish, these fishes will be around in and around area of the sea, our cage location only. We collect the fish, transport the fish in the boat with the help of FRP boat, and brought to the cage station. We'll give 200 ppm formalin for 1 hour, and then released in the cage. The grouper fish are having they are all called protogynous hermaphrodite animals. Initially they born as female. After 4 kilo or 4.5 kilo size, it changed to male. So we should have both size 1.5 kilo to 9 kilo size, so that we can have both male and female fishes. And similarly, and the other fish is called sea bass. It is protoandrous fish. The male is smaller, initially born with the male and is changed to female. This grouper is called protogynous hermaphrodite. So we should have the fish size range from 1.5 kg to 9 kg size. So after reaching the site, these are the cage maintenance. So every once in a month, we have to take out the fish, dip in the fresh water for 5 to 10 minutes to wade away the attachment of external parasites. And also at the same time, we have to change the dead cages to make the free flow of water. Because after 1 month, all the barnacles and other bivalves will get attached to the cages. And also we have to dive down to the cage bottom and check any damages, and we have to report. And feeding will be done once in alternative days. Why? Because if you feed every day, this fish will, because since the fish is in a confined area, there is no physical activity for the fish. So every day if you feed, this fish will eat, and more fat body will develop. It will affect the gonadal development. That is why we feed once in alternative days, and also it helps in digestion of the food. And we used to feed frozen fish like mackerel or sardine fish. The frozen fish initially it will not accept initially, because the wild catfish used to take the live fish only from the sea, from the wild. Whereas in the cage we cannot give always live fish. It's always, it may carry some pathogens. That is why we used to freeze the fish in the deep freezer -20 degree, and then we after thawing we feed the fish in every alternative days. After once the fish is accepting the frozen feed, then fish is almost conditioned. Then we have to check the maturity status, whether male or female is matured or not. How to check? The fish is very strong, very ferocious. We cannot handle the fish. Before checking the maturity status, we have to give anesthesia to sedate the fish. So we use anesthesia called aqueous. It's an imported product. Now it is available in India itself, and we use at the rate of 20 ppm to disorient the fish. Once the fish becomes sedated, we can do the maturation check. So before that, we have to tag the fish. Tagging means this is a radio frequency information with RFID tag. So this is a tag applicator. We have to fix this tag applicator inside the RFID equipment into the tag applicator and insert into the fish after anesthetizing the fish. After the tagging, and you have to go for maturation check. For the female, we have to insert a cannula for the female reproductive organ, and we have to take out the eggs, and the F diameter is more than 400 micron. Female is matured. Similarly, if you strip the abdomen oozes out the milt. If milt oozes out means male become matured. So once we get the matured male and matured female, the sex ratio is for one male, we have to have 3 females. So accordingly, we cannot put 2 males together, matured male together in single case, always fighting will be there. That's why 1 male, 3 females put in a separate cage. This is the arrangement we make. Mostly other food fishes, they go for induced spawning, whereas the groupers will spawn naturally. So we need not go for induced spawning. So what we will do, the grouper fishes are put in inside cages, and outside there is a protected net, and in between sandwiched with the 400 micron mesh. During night time, we just raise the 400 micron mesh and to collect the eggs after spawning. So next day early morning or night, the spawning will occur during midnight, and next day early morning we collect the eggs and pack it in the oxygenated bag and take it to the hatchery. So this is the egg with having single oil globule. It shows the good quality eggs. If it is having more than 2 oil globule, the eggs are not good. This is a grouper hatchery. This is the old finfish hatchery in Andaman. We took it on lease and modified for the grouper seed production. And we take a seawater intake system, the 25 meter depth from the open sea. And this is a seawater intake system and a water filtration system. So before doing this activity, we should have a live feed because initially for grouper larvae, live feed is very important. Rotifer and copepods. For production of rotifers, we should have a Nanochloropsis algae. So that system should be developed first before bringing the eggs into the hatchery. So we produce the Nanochloropsis in the hatchery laboratory, and to the mass scale. This is a microalgae strain room, small scale, and to increase to 20 liters carbide. From 20 liters carbide to intermediate carbide, from intermediate carbide to 2 ton tanks on the outer algae. This is a rotifer. This is a SS type rotifer. For the initial feeding, depends upon the mouth size of the fish. So these rotifers are grown in the rotifers are fed with the Nanochloropsis, and this before feeding, the rotifers are enriched with a commercial enrichment product called Algamac before feeding to the larvae. And these are all the copepods. This is called Paraconalus copepod. It is naturally available in Andaman. These copepods are naturally having PFA, polyunsaturated fatty acids, and it is very much helpful for the growing the larvae and very much suitable for the initial larval size, because naturally rich in essential PFA and well digested by the algae, and small size is suited for early life stages of the fish larvae. Swimming motion that triggers a feeding response for the larvae, because initially the fish will not swim that much fast. So the copepod napoli will go nearer to that mouth and easily it will help to catch. This is the stage we bring the egg to the hatchery, and we treat the egg with the ozone for 60 seconds to remove the pathogens. After the ozone treatment, we stock the eggs directly into the larval rearing tanks at the rate of 10 eggs per liter, and hatching takes place 18 to 20 hours at 28 to 30 degrees Celsius. The hatching rate, mostly 80% hatching rate will be there. The mouth opens after 48 to 56 hours. Once its mouth open, we immediately should feed with the SS type and copepod napoli. So the groupers, why the name came, because always they are all in groups. The larvae are in groups in the tanks. So to avoid the group formation, if it form in the groups, it will secrete a slime layer, the larvae will die. So to prevent that, we should maintain a homogeneous color in the tank. For that, we have to slowly drop the microalgae, nanochloropsis, to maintain the green color. This is the movie play avada. Next. Illa correct than. Next slide. அடுத்த slide. Click பண்ணுங்க. So movie is not playing. These are the different larval stages. Initially yolk stalk stage, and then initially feed stage, and swim bladder inflation stage. These are several larval stages. Once it reaches day 19-14, we have to feed with LS type rotifers. So all our movies and it's not playing. This is a day 21 larvae, and this is a day 35 larvae. This is a different feeding protocol for the tiger grouper larval rearing. These are the water quality to be maintained. For the temperature should be 28 to 30, salinity should be 30 to 34. Light is very important, and some 500 to 700 lux. So photo period should be natural. Photo period should be maintained. Feeding should dissolved oxygen should be 80 to 100% saturation. These are the things we have to maintain for the larval rearing. And after that, we have to go for nursery 1 and nursery 2 phase. Nursery 1 phase from 1.2 centimeter total length to 2.5 centimeter. It takes 2 weeks. Grading should be done. This is a nursery phase, second phase nursery. So this is a size of 10 to 15 centimeter total length for juveniles to stock in the cages. This is a stage, ideal stage for stocking in the cages. So during larval rearing, this type of deformities will occur, and this should be avoided by using the vitamin C and other things. And we did 4 farming trials. This is a market size fish for live fish market for the tiger grouper. This is after harvest of tiger grouper. These are all some during cage culture. This during rainy time, rainy season, this pop eye problem will come because of bacterial infection, and these abrasions will start because of when the seas become rough. So we have standardized technology for the grouper broodstock net cages and hatchery and nursery production of grouper fingerlings. In the cage culture of the hatchery and hatchery produced tiger grouper fingerlings in marketable size. So issues and challenges is that once the international air cargo is established from Andaman, this project will be a successful one, and also it will conserve the fish depletion, especially grouper fish depletion. Sorry, I could not play the movie because of some technical issue. Sorry. Thank you very much.
We sincerely thank Dr. Dinesh and the Lejuni Mrcvbr sir for his more informative and inspirational speech. I think all participants gain more information from his speech, so which has greatly enriched our understanding, sir. Once again, I thank you, sir.
It's a great pleasure and delighted to welcome Dr. K. Anandraja sir, who is currently serving as Senior Scientist in the Nutrition, Genetics and Biotechnology at ICAR, Central Institute of Brackish Water Aquaculture, Chennai. Dr. Anandraja sir completed his PhD in Aquaculture from ICAR, Central Institute of Fisheries Education, Mumbai in 2024 and obtained his MFSc in Aquaculture from Fisheries College and Research Institute, Tuticorin. His professional journey includes services at ICAR, Central Institute of Freshwater Aquaculture, Bhubaneswar, National Academy of Agricultural Research Management, Hyderabad, Department of Fisheries, Government of Tamil Nadu, and also as a fisheries science teacher in the Republic of Maldives. Currently, he is involved in several ICAR-funded research projects focusing on nutrient optimization, sustainable feed development, biofloc technology, and broodstock development in crab, shrimp, and fish. He has published 17 research papers, 5 book chapters, and has received Best Oral Presentation Awards at national and international conferences. His recent achievements include successful seed production of the critically endangered peninsular cod, Hypsilobarpus calculus, and standardization of biofloc-based rearing strategies for improved growth and survival. We are confident that his presence and expertise will greatly benefit all participants today. Welcome you, sir. Thank you.
A warm and respectful welcome to our keynote speaker, Dr. K. Anandraja sir. I kindly invite him to address the PPT presentation.
Good afternoon to all. Myself, Dr. K. Anandaraja, working as a senior scientist in ICAR SIVA, Chennai. So I will be presenting on topic recent development in brackish water aquaculture in India. So previous presenters and colleagues presented about the coastal region development and marine fisheries related aspects. So my presentation will be mostly on brackish water aquaculture. So aquaculture, you know very well, it is a culture of aquatic organisms in the water. So here brackish water aquaculture. What is brackish water aquaculture? So the you may know about different types of culture practices based on the water. Freshwater aquaculture, where you will culture the fishes in freshwater, where the water is 0 BPT, whereas brackish water, where in between 0 and 35, there is a brackish water, where whatever the fishes and shrimps and living in that area, that we will culture. And here we will discuss about the brackish water aquaculture. So brackish water aquaculture is a key driver in coastal blue economy, and India has significant brackish water aquaculture potential along 8,085 kilometer coastline. We have the vast coastline, 8,085 kilometer across the coastline. So the potential brackish water area is we have is 1.2 million hectare. Inland saline soil we have 8.62 million hectare, but at present only we are using only 1.28 lakh hectare area is under culture, brackish water aquaculture. So government is aiming to bring additional 1 lakh hectare area under aquaculture. So now we stand globally a second largest producer, aquaculture producer in the world. And shrimp farming, if you see the brackish water aquaculture, the major species cultured is shrimp. So shrimp, especially Litopinus vanami. So shrimp farming is the dominant activity in brackish water aquaculture, contributing to the bulk production of bulk of the production and export. Production is increasingly intensive, commercial and market oriented. Previously, in olden days, it is a traditional culture. Okay, only simply they will stock the seeds or coastal farms, whatever the tidal farms, tidal entry of fishes, that will be stocked in the pond and it will be grown. Now, it is mostly of commercial scale. Okay. So the world fisheries and aquaculture production has hit a new high in 2022 with aquaculture production surpassing the capture fisheries in the first time. If you see the production trend, total production world fisheries, it is 110 million tons. Now it is 185 million tons in 2022. Okay. So if you see the aquaculture production, it is steadily increasing from 1990s 21.8 million tons to now 94.4 million tons. Whereas capture fisheries almost stagnant. You can see 88.9 million tons to 91 million tons only. So aquaculture production is steadily increasing based on the demand. So major species cultured in brackish water aquaculture is shrimps, especially Pinnaeus vanami. The principal species cultured in this is Pinnaeus vanami and Pinnaeus modern. Still we are culturing. Previously it was Pinnaeus modern mostly cultured, but now Pinnaeus vanami is dominating, and it is in selectively it is cultured. Nowadays farmers are also culturing Pinnaeus modern also. Other than that, finfishes, Asian sea bass and mullet milkfish also cultured. So nowadays many farmers facing a disease outbreak in the shrimp farming, so they try to switch over other species also. So like mud crab, Scylla serrata, it is 1 kg is around 2,000 rupees in the market. So people are, farmers are trying to culture this crab cultures also, and species diversification promoted due to risk, reduced risk and enhanced sustainability. Okay. So these are the common species that we are doing the work. Lattice calcarifer and Heteroplus rotensis, chanas, and pyflock based shrimp farming for Pinnaeus indicus. So other than that, in finfishes we are producing seed production and farming of Asian sea bass. So we have developed the technology for this, and we have indigenous feed formulation for maturations also. So sea bass can be cultured in both in earthen ponds and brackish water open sea cages. It is a urigalon fish. It can be cultured in 0 to 40 ppt. We have signed a MOU of 10 MOUs in providing consultancy and technology transfer to farmers and hatchery operators and entrepreneurs and other clients of sea farming. So this is another one group of fish. This is a modular system-based seed production technology for pale spot, Heteroplasmus frutensis. So it is easily can be it can be easily adopted by small and medium scale aqua farmers and self-help groups. And low input cost technology. Regular income can be generated as a part-time activity also. This is filed for patent also, this technology, seed production technology. And you can see generally this Scylla serrata and crabs, no, they are cultured with trace fish. Whatever available in the local market, farmers they collect and they feed the fish. Now we have the feed for crabs also. Scylla serrata and Porcinus pelagicus also we have the feed. They are readily accepting the feed, and growth also similar to the trace fish. Okay. So molting weight can little bit higher in trace fish, but it was a little bit lower side, but it is at par with the trace fish feeding. So in upcoming years we will come with this Scylla serrata and Porcinus pelagicus feed also. So these are the major area we work, and we have developed technologies. So in hatchery seed production we have Asian sea bass, milkfish, pale spot, and long wilker catfish, silver moni, spotted cat, mangrove red snapper, and Indian white shrimp, and mud crab. This technology we have. And farming technology we have culture of Indian white shrimp, biofloc technology for shrimp nursery and pre-growth seed rearing methods, and cage culture nursery and pre-growth, and polyculture technology for freshwater finfishes, production of marine polychaete and Mrcvbr, and integrated multitropic aquaculture, and super intensive precision and natural shrimp farming. This super intensive and natural shrimp farming, it is very successful one, and we have received a lot of appreciation and awards also for this super intensive farming. I will show you the video also in upcoming slides. And feed technology, we have indigenous shrimp feed processing, and shrimp larvae plus, Vanami plus for growth, and shrimp larvae plus, hetero brood plus, milkfish brood plus, poly plus for polyculture, and color fish for ornamental fish, and Scylla plus for mud crab, and plankton plus for plankton booster from the fish waste. And other than that, we have the products like Sipamox. It is a water probiotic for controlling ammonia, and a nested RT-PCR kit for WSSP, nested RT-PCR kit for EHP, and a nested and RT-PCR for VNN, Lumipage, Bacteriopage, it is for Vibrio, and parasiticide, antiparasitic vaccine for VNN, and EHP Cura. You might have heard about this product. It is successfully moving, and many farmers and many places we demonstrated across India, and PCR kit for Vibrio Harvey, and similarly shrimp crop insurance. So these are the most common area we work, and we have the package of practices. And some of the farming system innovations I will show you here. There previously it was extensive culture or semi-intensive culture. Now most of the farmers are switching towards super intensive farming, and based on the lined pond or lined systems. And now biofloc also upcoming, and it is a zero or minimal water exchange system. And introduction to biofloc and microbial based culture techniques, and use of higher stocking density with better management practice, and easy initiative towards integrated and diversity farming system models also we have developed. And this is the biofloc based shrimp farming technology. Here what is the concept is that when you feed the fish, fishes consume the feed. Some portion of fishes feeds are not eaten, and some portion of the fish whatever eaten, it is coming as out as a excreta. So the nitrogen is loaded in the system, and during the heterotrophic bacterial bloom, what happens is that it takes the nitrogen from the system if you provide the carbon source. C-N ratio, if you maintain 10:1 to 20:1 in this ratio, in between if you maintain, by adding carbon source, the heterotrophic bacteria will absorb and multiply by using the nitrogen source present in the system, using the carbon source we provide. So automatically there will be a bloom of heterotrophic bacteria that can be used, there will be a food chain formation. So automatically the waste feed or waste content also it will be used twice, okay, recycled. So this is the concept of biofloc, whereas in aquaponics it is somewhat different. It is autotrophic based system, nitrosomonas, nitrofactor based culture system. Okay. So this is a zero water, minimum water exchange system. Okay. And see, future must, as per aquaculture, Sophia book, future aquaculture expansion must prioritize sustainability. Whatever work we do, it must prioritize sustainability. And biofloc technology is a sustainable production tool for aquaculture. The main concept is we have to maintain the CN ratio. Once we maintain the CN ratio, there will be a heterotropy bacterial bloom. Automatically, whatever the nitrogen waste is there, it will be assimilated from the water. So, what is biofloc? Biofloc is composed of biomass of bacteria, fungi, flagellates, protozoans, ciliates, algae, and other microorganisms, extracellular polymeric substances, and organic matrix. It will be in the particulate form, where these organisms will be also there. Okay? So, to respond to growing demand of animal protein, water, and land resource constraints, the intensive aquaculture like PVT is one of the options. So, importance of carbon sources and CN ratio. Most of the carbon source, there are many types of carbon source they use. Mostly molasses and jaggery, it works well. It is readily soluble and available, become more available. Carbon source will be readily available. Whereas other carbon source like flour of wheat flour, rice flour, it takes time, but it is also working, and it is in experimental level only. Other carbon source used in the PFT are sucrose, dextrose, glycerol, and glucose and acetate also they are tried it. And beneficial effect, if you see, there are a lot of microorganisms in the biofloc. So that promote the growth, that act as a probiotics. Okay? So biosecurity of the system can be maintained since there is no water exchange or recirculation. So once we have loaded the water in the system and we maintain the biofloc, so it will be in the confined environment. There is biosecurity is maintained. So, second is heterotropy bacteria can reduce toxic metabolites. It can assimilate also, it can transform the metabolites. So it can be heterotrophic bacteria can reduce the toxic metabolites. Doubling the protein utilization as the shrimp use protein twice, eat feed other than and then harvest the flax. Okay? Enhance the digestion. Even the flax contains that organisms contain digestive enzymes and growth promoters that supports the growth. So probiotic actions also there, and immune response also there. And one of the major advantage of this is that no specific system for this. Only is common simple lined pond or tanks we can do. Only we have to extra cost we have to provide the aeration for this, and carbon source also we have to provide. Okay. And digital and smart aquaculture. So nowadays farmers shifting towards the digital and smart aquaculture. Nowadays farmers also in the field they have the sensors. Okay. They are 24-hour automated sensors also there. So DO, if it comes below 5, it will give the alert. Directly you can act. Okay. So the DO, pH, temperature sensors also there, and automatic feeders also there. So we have also developed super intensive farming in that in our Muttugadu experimental station. I will show you that system. So this is a super intensive precision and natural farming system. It is with improved productivity and profits in controlled environment, improved input use efficiency such as land and water feed, minimal risk for disease and climate change. ISA-SIFA has signed 5 MUs. It is Andhra Pradesh, Tamil Nadu, Maharashtra to set up this farming unit. Here the concept is we have one central nursery and 4 grow tanks. It is like nursery rearing, 1 month we will rear. Once it reach 1 gram, PL13 to 1 gram, then we will shift to grow tanks. Okay? So it will be year-round production is possible, and more than 100 tons per hectare per year it is possible. Whereas in normal earthen pond culture, we have only 5 tons, 3 to 5 tons per hectare per crop we will have. But from here, 2 tons per crop per 3 months we will get it here. Okay? So this kind of production is also there. I will show you the video of this super intensive farming system. It is in ISRCPA Muttugadu experimental station. Okay? Dr. Kumaraguru is the principal scientist. Sir has developed this system, and I will play the video. I request to play the video. Super intensive system. This is the superannuation recirculatory system. Now we have gone stocking density of 360 per meter cube. The production is 2.25 tons per tank. We have reached that FCR is 1.16. It is the lowest. Cost of production comes to 176 rupees. You can see the 4 crowd tanks with a central nursery. Okay, this is about our superintensive system. So we have received a lot of appreciation from Government of India and a lot of awards also received by Dr. Kumaragur sir. The second, this is a production system we have successfully made in the coastline of in the Muktugad experimental station. Other important area that we work is genetic improvement program. Nowadays, everywhere if you see culture in the culture pond, you can see the Lithopenes vanami. Now we are working on Ipnes indicus. It is our own indigenous species, indigenous shrimp. So we are in the process of developing our own variety that is growing fast by selective breeding program. So we have initiated that program. I request that to play that video, next video. Genetic improvement program of Pinnaeus indicus that we work on.
Panite shrimps are the most traded seafood commodities globally. India contributes 0.9 million tons worth USD $5.8 billion to global shrimp production. Scientific shrimp farming in India started in early 1980s, dominated by black tiger shrimp, Penaeus monodum, and Indian white shrimp, Penaeus indicus. However, emergence of lethal viral diseases like WSSV urged the industry to call for a new blueprint for shrimp farming based on genetically improved stock and selective breeding programs. Currently, Indian shrimp farming is dominated by SPF Penaeus vanami, although SPF Penaeus vanami introduction gave much needed impetus to shrimp industry. Overdependence of single species with emerging diseases and escalating price of brood stock calls for species diversification. Penaeus indicus is selected as a species for diversification with the national-wide demonstrations by SIBA funded by NFDB. It is recorded that Penaeus indicus has got huge potential for commercial or scientific farming with a production of 3 to 7 tons per hectare. Comparable with Vanamai, even without genetic improvement. Under Pradhan Mantri Matsya Sampada Yojana, PMMSY, Government of India has sanctioned the nationally important project to SIBA to develop genetically improved Penaeus indicus.
That was the GP
With the
program
dedicated
site.
scientific team and R&D efforts, a state-of-the-art experimental biosecured shrimp hatchery was
These are our
established
indoor
with 10
facility
million capacity
for
in a year,
development
with primary
program.
quarantine section to screen wild-sourced broodstock, improved biosecurity hatchery management practices. The scientific team of SIBA made significant achievements in improving the reproductive performance of native shrimps, captive broodstock development, and addressing the inherent reproductive challenges through innovative techniques like artificial insemination, single pair mating, environmental Q manipulation, etc. ICAR, SIBA, has decoded the complete genome of Penaeus indicus, first in the world to have sequenced a complete genome of penile shrimp. Concerted research effort on stock characterization for genetic improvement program is also being carried out for genetically improved Indian white shrimp production. An improved and SPF Penaeus indicus could serve as better alternative and a complementary species to Indian conditions of farming. This program will lead Indian aquaculture towards indigenous technologies to become Atmanirbhar Bharat.
Okay, now I have shown the videos of two major important programs that we are working on now. So many stocks of Penaeus indicus we brought from different locations in India, and few stocks are performing well. At present, we are breeding the Penaeus indicus, and we are distributing the farmers also. Slides. Okay, this is about the super intensive precision farming, and technologically development in practical agriculture. If you see adaptation of specific pathogen-free broodstock, especially for shrimp, it is strictly followed. That's why we are able to sustain the disease outbreak when other countries faced. In long back 2013-14, most of the countries had the disease outbreak, but we sustained. Even shrimp market, international buyers came to Andhra, and shrimp prices gone to 1,200 rupees also, I remember, when I was surveying the Andhra Pradesh. So that much strict guidelines is followed, and even RGCA involved in that quarantine facility, maintaining the strict, following the strict guideline and supplying the broodstock to hatcheries. Okay, so we are able to follow that. And emphasis on standard operating procedure for farming operations. So this is must be it is followed, and advances in formulated feeds with higher feed efficiency and reduced FCR. So if you see the production, any aquatic species, either shrimps or crabs or carps, almost 50 to 60% of the production cost is feed cost. So feed efficiency must be maintained so that your production cost of the species will come down. So I will show you the different types of feeds that we are producing in our SIBA. So this is our own feed mill. You can see that there is a hammer mill here, pulverizer, and other than that, after making all the ingredients, we have a lot of different types of ingredients. Fish meal, it is one of the protein source. Soybean meal, it is a plant protein source. And fish oil, it is a lipid source. And carbohydrate, we use wheat flour, rice flour, and vitamins, minerals. So different types of ingredients we have. All over world, 200 types of fish feed ingredients are available, and based on the locality, based on the availability, based on the digestibility and the nutritional value, we can make indigenous feed formulation. We have the technical know-how. Dr. Ambasankar sir is there, our head of division. is very expert. Sir is having very expertise in this formulation based on the nutritional value of the ingredient. And we have the feed for all variety of fin fishes, whatever I have shown, and for different stages. So this is one of the most successful larval feed. This is shrimp larvae plus. Okay, this is a cost-effective indigenous larval feed for superior quality. So it contains 55% crude protein and 12% lipid. It is the ingredient with high digestibility. It has EP and DHA also. Here we produce for internal our use also, and commercially we are giving to Andhra Pradesh. Many hatcheries and business owners, they are requesting us for supplying the feed. Okay, this is cost effective and functional feed for shrimp larvae. We have signed many MOUs in Andhra Pradesh and Tamil Nadu. And Vanami plus shrimp feed. Our feed cost is only 90 rupees. And if you go to the market, it is very high cost. It's because we have the indigenous formulation, we can develop our own feed based on the locally available ingredient, what is the nutritional value and the digestibility. So we can, if the particular ingredient cost is going up, we can reduce that particular ingredient, and we can alternative ingredient we can choose, and we can reduce the cost, we can make the feed very cost effective. Okay. So it is made in the pelletizer. There are two types of machines available for making feed. One is extruder, another one is pelletizer. In the pelletizer, the feed blend is compressed. First step is you are choosing the ingredient, then you are making based on the formulation, then you are grinding and you are making pulverize it. 100 micron size particle like maida powder you are making, then you are loading it in the pelletizer. It will compress that flour into pellet form. What I am showing, that pellet like. It will be like a small pellets. Based on the mouth size of the fish or shrimp, we can make the pellets from 300 microns to up to 11 mm size we can make the pellets. We have the feed mill capacity like that. Okay. So commercial scale feed cost is very high. We are having only 90 rupees. Whatever super intensive production, that is 2 tons per 500 meter cube tanks we have produced. All the shrimps production, it is based on our feed mill feed produced. That is Vanami plus shrimp feed only. The another feed we have Seabass plus Seabass feed. So it is available in slow sinking and floating feed also. In the floating feed we make it in the wet extruder, whereas sinking feed in the dry extruder. Okay. So this feed is also commercially successful, and we SAC demonstration we are giving, DSP demonstration also we are giving this feed. And color filies, it is an ornamental fish feed. Okay. This is available, it is also available. Many MOUs are signed. Women Entrepreneur Haryana and Startup Chennai, they have taken the technology. And plankton waste to wealth. This is one of the unique one. Here what happens, many places in the fish market and many areas, this fish waste is there. This waste will be collected and it can be used as a fertilizers also. So we have the technology, indigenous technology, and we have the analytical reagent. By adding that waste, whatever the offal, fish offals will be converted into plankton plus liquid that will be collected and packed, and it will be given to the farmers. Okay. And health management, mostly disease outbreak have saved modern practice of agriculture practices. Mainly that biosecurity protocol. Many farmers are following biosecurity protocol nowadays. Any farm if you go, they will have the bad fencing because white spot disease will come. So bats will spread the diseases. So that like that many use of probiotic, immunostimulants and functional feeds. And nowadays farmers also aware that antibiotics they should avoid. Okay. So implement of pest management practices across the farming clusters. These are about this. And development of innovative aquaculture products. So these are the products we have in our SIBA. So this is SIBA MOX. It is a water probiotic. Yokita. It will reduce the ammonia. When you feed the fish, some part of fish are going down and it will be disintegrating. Ammonia will be developing. Akshari. This is a SIBA MOX. It will control the ammonia. Ambika. Okay. And this is a viral nervous necrosis vaccine. So for food stock development,
5 minutes.
Vaccine developed, and it is very effective compared to getting vaccine from outside. Then SIBA natural phytochemical and nutritional supplements that significantly reduce the EHP and withdrawal load. It improves the gut health. Growth of the shrimp can reduce the usage of antibiotic and compound chemicals. Multi-parameter water quality kits also we have. Water quality parameters we can measure easily. And we have the improved PCR kits for reduction of white cell syndrome virus. And the real-time PCR kits reduction for EHP. And we have the patents, 19 patents filed. Out of that 13 granted. And the copyright 18 and granted 2, trademark 4 and granted 2. Ours are in the line. These are the patents. And we have the ISO certified technology. We have
मध्ये ब्रह्मणे नमः दक्षिणे नंदाय उत्तरे सोमाय मध्ये रुद्राय अष्टमे विष्णवे बले इंद्राय नमः सत्यं धर्चे उभय त्रिशूल सुंदर सुंदर भगवान महाराज नमः मध्ये सुलभ चे पूर्वाग्र उत्तराग्र कुशलपदेन धुष्पदर्भा ओम यम अवने नमः सुदोदाम नमः नमः मोजना मोजास्त्राय नमः कुंडस्य भितरे
many aspects of research institution driving the research, technology development and capacity building. About policy support and institutional and the challenges. What are the even some climate variation affect the breeding program also, our own breeding program also. So in MICRO project we have created our own system.
We're a software consultant.
Whatever the environment required for the breeding of the fish and shrimp. Okay. So environmental and social concerns related to the fish and land. So the aquaculture, whatever the culture, whatever should be maintained. Issues, concerns also, it is a challenge for us. And a limited diversification leading to shrimp dependence. So till date, most of the farmers depends upon this shrimp, Lithopenes vanami only. So switching over to crabs and other seaweed-based polyculture, so other shrimp indigenous variety, it will make more, it will alleviate the problem of shrimp dominance. Okay. Way forward. What we have to do is strengthen the sustainable intensification through smart and best aquaculture practices and scale up selective breeding and genetic improvement program for indigenous species and enhance biosecurity hatchery technology and to ensure the continuous food stock and gamete availability, promote diversification aligned with the market potential. Whenever we go for diversification, we should see the potential of the species also. Farmers, how can we our stakeholder, we can fulfill the needs of our stakeholder. That also one point we should take care. And expand integrated multi-tropic aquaculture and cage culture based livelihood models for coastal communities, and optimize feed efficiency and cost effectiveness, waste to wealth and circular economy approach for value addition. So we have a lot of resources of inland saline. That also we have to work on, support inland saline aquaculture with region specific technology and the sustainability assessment. We have to assess first, because even though it is a saline affected soil, we should assess. Without assessment report directly we cannot go. So how far a sustainable way we can promote this inland saline, how can we use, that have to be promoted, and prioritize nutritional security, domestic consumption and alternative livelihood alongside export. So domestic consumption also we have to promote, because if recently there is anti-dumping duty and banning of shrimp import from the USA, so many countries may be affected. So we should promote our own domestic consumption also, so that cannot that we can withstand this kind of bad weather. Okay. So in conclusion, what we can say is that brackish water aquaculture is the key pillar of Indian blue economy. Sustainable intensification of brackish water aquaculture is critical for global seafood supply, nutritional security, livelihood and export earnings. Emerging challenges demand synergic action between research, policy and industry. And what we need is environment friendly and climate resilient practices and productive gains through domesticated improved varieties. Already we are working on. So disease diversification and disease resistant farming system and science-based innovation, selective breeding program and timely stakeholder adoption will determine the future growth. Not only developing technology, we have to transfer the technology to the farmers, and it must be adopted. And whatever the issues facing in the field level, that again back to lab level, and it has to be addressed. That kind of flows must be there. And MICROBIologY plays a central role in technology development and policy governance and sectoral transformation. These are the recent development in brackish water aquaculture. So with this I conclude my presentation. I thank our institute director, MICROBIologY director, Dr. Kuldeep Lal sir, for providing this opportunity to present in front of you as a keynote speaker. And thank you.
Thank you, Dr. K. Anandaraja sir. This is very more useful session. I think participation gain knowledge about SIFA commercial products. So this is really very useful to us. Once again, I thank you, sir. Thank you, sir. Next, I warmly welcome Dr. Sujit Kumar Patnaik, sir, Junior Fisheries Scientist, Ministry of Fisheries, Animal Husbandry and Dairying, Government of India, Mumbai. Sir, we welcome you, sir. So now I hand over the sessions to our keynote speaker, sir. Sir, please present your keynote address, sir. Welcome, sir.
We are honored to introduce Dr. Sujit Kumar Patnaik sir, who is currently serving as a junior fisheries scientist at the Fisheries Survey of India, Kochi, since June 2021. Dr. Patnaik sir has a strong academic background with an MSc in Marine Science from Berhampur University and a PhD from Andhra University, Visakhapatnam. In addition, he has completed postgraduate diplomas in Agriculture Extension Management and Computer Applications, which greatly support his research and outreach activities. He has been associated with the Fisheries Survey of India since 2002 and has gained wide professional experience through his work with ISRO's OceanSat-1 project and the National Fisheries Development Board. He has actively participated in several fisheries survey cruises, including Equatorial Water Survey for albacore tuna. His key area of work include marine fish resource assessment, exploratory fishing surveys, remote sensing applications in fisheries, data management, and sustainable fishery management. He is also involved in capacity building, training, and academic guidance for fishery students. With nearly 30 research publications in national and international journals, Dr. Patnaik sir continues to make valuable contribution to marine fisheries science. We are confident that his experience and insights will greatly enrich today's program. With great respect and warm regards, we welcome Dr. Suchit Kumar Patnaik, sir. Welcome you, sir. Thank you.
Good afternoon. Vanakkam. Namaskar. So, before I start, I must beg pardon from the organizers because my presentation was actually planned for yesterday, but due to some unfortunate official assignment, I could not participate. So I requested the organizing secretary, Dr. Dinesh sir, to just postpone my PPT for tomorrow. So thank you so much, Dinesh sir, and all the organizing committee and all the members. So my presentation is a little different. It's not like others. Means in my presentation, when I was told that most of the participants will be students, so I just changed my mind. I thought that what should be more interesting for students, though they are the future of the nation. So what are the things we can create among the students by which they should not feel bored, and in the same time they will carry forward the same awareness among all the people. So my topic what I have kept is marine fishery resources of India with special reference to deep sea fishery resources. So what are the scopes or opportunities and what are the challenges by which we have to come across so as to contribute to the blue economy of our country as expected by the Honorable Prime Minister Sri Narendra Modi ji, to have India the the powerful nation in the world by 2047. So to just take forward his mission and all the Government of India organizations are taking lot of initiatives, lot of new technology development, everything they are doing. So my topic I have taken little different. So before that, when I had a small discussion with your principal sir, so what he told, our students, everybody is talking about the general public. Okay, public actually contribute equally to the nation building, but the students, if they are well aware of many things, they can create magics. So what he requested, just to have little bit awareness among sir pollution, because now pollution is a bigger threat for all the nation, even a developed nation and developing nation like India. So just 1 minute I will take. In global level, Glow Litter project has been initiated by almost all the countries just to eradicate all the different type of pollutants from the sea, because sea is the main area where wherever you throw any plastic, especially plastic, I am talking about plastic, wherever you throw, definitely in through the runoffs, through the drainage lines, it will finally come and reach to the ocean. So the ocean getting day by day fully polluted. So I request all the students not to indulge yourself in these activities. Rather, you should create awareness among all the people who come across not to do such things. Plastic use cannot be stopped, but it can be managed. How? Just we follow all the guidelines, whatever is given. Don't throw here and there. Put it in the assigned garbage bag or in the dustbins. Okay, such awareness you have to create. Okay, so before going to my presentation, just I would like to tell something about my organization, because you all are students and your principal sir was asking, sir, whether fisheries has a future prospects. I told definitely. Not only Government of India, all the state government and Government of India is taking all initiative to ensure that the fishery sector can contribute maximum to the gross domestic product. of this country, GDP of the country. So almost, as sir told correctly, 20,050 crore rupees under PMMSY, Prime Minister's Matsya Sampada Yojana, under my ministry, has been given to all the stakeholders so as to promote and to ensure better a contribution from fisheries to the GDP of the nation. So, just next. Yes. Just 2-3 slides I have made about my organization. The name of my organization is Fisheries Survey of India, and this is one of the most oldest fisheries organization in the country. It was established pre-independent because our country got independence in 1947, whereas our department was established immediately after the Second World War. Okay, why? Because the entire world is fighting for starvation. The food security was the major threat after the Second World War. So that time it was decided that to have a natural protein source of food, that is fish, sea fish. For that, in 1946, it was established as offshore fishing station. The name was different. But slowly, it was more and more objectives, more and more mandates has been added to the department. And in 1981, it was renamed as Fisheries Survey of India, and it was recognized under Department of Science and Technology. And
எங்களையெல்லாம் செதுக்கிக் கொண்டிருக்கும் நடுவர் பெருமக்களுக்கும் சிற்பங்களை போல் அமர்ந்திருக்கும் நாளைய இந்தியாவின் அரசு
husband in dairy, New Delhi.
மணியொலி கேட்கும் ஒரு பெல் ரிங்.
In Mumbai, of course, I am working in Kochi, Kerala. And we have 7 operational bases throughout the Indian coast, starting from Veerable, Maharashtra in Bombay, in Goa, in Kochi, in Chennai, in Visakhapatnam, and in Port Blair. And our main mandate, what FSI is doing, everybody should know, because the government is not paying to the employees from their pockets. It is the public money, and public has the right to know what government is doing. Okay, so you should know different, so many organizations have been formed by the government for different objectives, for different mandates. So our main mandate is resource assessment. That is survey and assessment of fish stock and charting of fishing grounds in the Indian EEZ. EEZ, I think everybody must be aware of what is EEZ, exclusive economic zone. Okay. And monitoring of all the fishery resources, gear suitability. Gear means the net, it may be a net, it may be a line, anything to catch the fish is called as a gear. So gear suitability, for what type of resources, what type of gear is suitable. Then come forecasting and remote sensing. Use of use of remote sensing in fisheries. How we will forecast? Means what type of fish, where it is available, so that the fishermen can easily access and catch the fish. Just to reduce the expenditure or to reduce the pollution also, you can say. Because once you run a boat, a motorized boat, you have to just chase behind the resources. But once you know that where is the stock is available, so easily you can go and catch the fish. So that also one of our work. One is data dissemination, means whatever data is scientific, after the scientific survey, what are the scientific findings we have, how, in which way that can be disseminated to the end users. So who are the end users? Obviously the fisher community, and of course the researchers and the scientists. Next is our human resource development. So as sir told, our department also recognized as a research institute for leading to MSc and PhD. So if once you register in an university, almost in all university our department is recognized as a research and a scientific department, where any student can register in the university, he can come to our office in our lab, he can work, pursue, he need not to go to his college. And after that he can appear the exam and he can pass. Yeah, likewise, you can pursue your PhD also. Still so many, around 20-25 people, students there pursuing their PhD degree. Because almost all scientists are the recognized co-guides in almost all university wherever our base is located. So as I told you, we have our presence all along the Indian coast. Already I have told. And these are the type of fishing gears our department has been deploying. All the type of gear. This is trawling. This is pelagic trawling.
Here,
that is
juries will
trap
be sitting in
fishing.
front of you,
This is long
and
lining.
the judgment will start.
This is vertical long lining, and that is squid jig.
What if your computer could be in two states at once? Meet the qubit, the building block of quantum computing.
But presently, we are deploying this much gears. That is why I have shown you. After Indian Navy and Indian Coast Guard, this is the only department, means my department is the only department who is
I extend a
having
warm welcome to
almost 17 survey fleets all over India.
Meaningful
17
when you
survey
secure the
fleets. And these are the two which are located in Kochi. So just I have shown it. Likewise,
Anamika,
17 survey vessels we have. After
Kaushika,
Indian Navy and Coast Guard, you will find Fishery Survey of India has the largest
Lavanya,
fleet size after these two Indian Coast Guard and Indian Navy.
Logapriya, Logeshwari.
So just as I am from a marine fishery background, just I already such things have been shown by many of the people. Indian exclusive economic zone is nothing but every country has their own EEZ. EEZ is nothing but the right, the sovereign right of the country to do any kind of activity. It may be a fishing, maybe oil spill, maybe a mineral extraction, whatever thing you can do within this EEZ. That is why it is called as exclusive economic zone. And this zone is confined with 2.02 million square kilometer. That is the area all around the Indian subcontinent. And the coastline, actually the coastline was earlier 8118 kilometer. But just recently, last year, after the survey carried out by National Hydrography Organization, NHO, and a survey of India, they told that earlier our conventional methods they used to measure the coastline, whereas with the upgraded technology, the new technology they used, they found that the coastline actually is 11,098 kilometer, whereas it was mentioned as 8118 kilometer. So our coastline has been what? Increased. Okay. So next is continental shelf, continental slope. These are very common things. I need not to tell again. The area of continental shelf in the Indian EEZ is 0.53 million square kilometer, whereas the present, as per the 2018 revalidation committee, the marine fisheries potential has been calculated as 5.3 million metric tons. And out of that 5.31 million tons, you see the pelagic resources. Pelagic resources is nothing but the midwater resources. It may be on the surface, maybe in the middle of the seawater. And demersal are the bottom dwelling, the fishes which usually inhabit in the bottom of the sea. They are bottom dwelling. Those are called as demersal resources. So their potential is 2.30 million metric tons. And pelagic resources is 2.63 million tons. And oceanic resources, that is deeper water, deep sea resources, that is 0.22 million metric tons. So these are the potential breakup of 5.31 million metric tons. And this is the last 3 years fish production. You can see why I shown all these things, because the coastal fisheries has been stagnant and it is under tremendous pressure. Everybody is going and catching fish from the near shore. So there, if we are not properly taking any management measures, after few days our future generation may not get fish. To just come out of this problem, Government of India is trying to diversification of fishing methods. Like from coastal water, you can go towards the oceanic or the deeper water, where plenty of resources are available which are still untapped. Untapped means which are not been cut. Simply the other country, they are just entering to our EZ and they are catching and going. But our people are not going, because they want money. For getting money, wherever the fish is there, you go, catch it and earn money. But earning money is not everything. Earning money, in the same time also you should ensure that you are leaving something for your future. If everything you consume, what the future generation will do? They will not get any fish. They will not get a natural protein. So my presentation is only to tell you that deep sea resources are plenty which need to be tapped or which need to be exploited, and Government of India giving lot of support by providing a subsidy, by introducing lot of schemes for diversification of fishing methods and fishing fleet, means vessels. Got my point? So the next 22, 23, 24. See, the potential was shown as 5.31 million metric ton, right? Whereas 22, the production was 3.49 million metric ton. 23, little increased to 3.53. Again in 24, it is 3.47. Why it is little less in 24? Because of the climatic impact. During 24, lot of cyclones and bad weather or adverse weather conditions, the fishermen, they could not venture into the sea, so the fishing could be hampered. So that is the reason the production is less. Next is, among the landings, see the pelagic, demersal and oceanic or deep sea. Deep sea is very meager, so it was included with the pelagic finfish. This is a breakup of 3.49 million metric ton is how it is done. From pelagic 1.99, which is 57%. From demersal it is 0.94, which is 27%. And crustaceans and mollusks and all those things are very a little percentage. So deep sea. So why I am showing all this, slowly we will go to the deeper ocean. Why deeper ocean we are so worried about? Why we are always promoting to deep sea fishery? So here just I am showing, deep sea is nothing but depth at which sunlight significantly diminishes. Of course, light has its own penetrating power. You know that you all are science students, you must be knowing reflection, refraction. Reflection is straightly entering and going inside. Whereas refraction, after entering from one medium to other medium, the light bends, right? So, if that process you see, the sunlight has its limitation. Slowly you go deeper, that means there will not be any sunlight. After that only it is a black sea. Up to certain length or the depth where the sunlight is penetrating, up to that little light will be there. After that no light, completely dark. So that deep sea means in that dark sea, lot of marine fishery resources are there, which is not being tapped. Tapped means caught. So that we need to promote. So deep sea fisheries, as I told, continental shelf and continental slope is well known to everyone. Shelf is up to the depth of 200 meter. After that there is a immediate steep slope where the depth of the sea very fast increases. Okay, that is slope area. Continental shelf is till it is 200-meter depth. So now everyone is promoting or targeting the deep sea resources which are available beyond 200-meter depth area. Okay. Means what is the uniqueness of the species? They are long-lived due to very less light, not much temperature variation. So their growth is very slow. See, for growth you need to have light and all oxygen, lot of things you need to grow. But in deeper ocean, the growth is very slow. Once the growth is slow, definitely their maturation also is slow. So they live for a long period. They are simply living for a long period and they are dying their own. Means natural mortality, it is happening. Whereas the catching mortality is not at all there. So why to waste the resources? It has a lot of, it can contribute tremendously to the blue economy. For that, we need to explore to the deeper ocean. The importance of the resources is just to reduce, as I told you, the coastal fishing pressure is tremendous. Everybody is catching fish in the coastal water, whereas nobody is going to the deeper water. So the pressure is tremendous. So the day will come, there will not be much fish in the sea. Just to reduce the pressure in the coastal area, just we need to go for the deep sea resources. And the significance is it play a vital role in food security and provide natural protein, as I told you, and also may contribute significantly to the developing blue economy strategy of Government of India. So what are the deep sea fisheries resources? Just very fast I am going through. Diverse marine life. So why it is diverse marine life? Because it's a huge area, complete dark area, encompasses a wide array of fish and shellfish species found exclusively in the deep water, adapting to low light, high pressure and cold temperature. No much temperature variation will be there. Till the light passes through the water column, only temperature will be slightly changed. After that, there is no temperature variation. It will be very chilled water. In that condition also, so many fishes are adapted to that area. And it is categorized species. They include demersal is there, benthopelagic is there, pelagic is there, and they have their unique role of harvesting consideration. They have their own ecological system within that. And they are underexplored. The potential means they have not at all exploited. They are untapped. Simply the resources are plenty in the deep sea. Nobody is catching. If we catch the fishes, we can contribute little, not little more to the blue economy of our country. And not only like that, it will create lot of employment opportunities. More resources are there, government need to have more infrastructure. If more infrastructures are there, definitely the young people, the educated people, they will get a chance to work with the new system. These are distribution of deep sea resources. As I told, continental slope and rise. Sea mounds means small mountains in the deeper water are called as sea mounds. Mountains, sea mountains means sea mountains, small mountains are there, it may be small or big, which are under the water, submerged condition. They are called as sea mounds. That is the very good habitat of deep sea resources because they are getting some shelter, right? When you go, all of a sudden you saw that heavily raining. If you find some area, some houses are there, you are not entering, you are entering to the house. Likewise, the fishes also, it's a natural shelter for the deep sea resources, sea mount. And beyond EZ means, as I told, 200 nautical miles from the shore. That is the EZ length you can see. From the shore, if you measure 200 nautical miles towards the sea, that is the EZ area. And beyond that is called as high sea. That is open for everyone. Everybody can go and catch fish beyond EZ. That is open for all. But our EZ, nobody else can enter without our permission. And other countries' EZ, we cannot enter without their permission. Okay, these are the constraints and these are the limitations. And Indian Ocean has its tremendous potential. Almost mesopelagic resources are there, oceanic resources. Mesopelagic resources are nothing but myctophytes. Whether you know or you don't know, I'm not sure. Myctophytes, just I will show you. Already those things I have told. And actually these are the conventional oceanic resources, which are 238, means 2,30,832 tons. That is the potential of conventional deep sea resources like tunas, billfishes, sharks. Okay. Whereas non-conventional deep sea resources like myctophytes, deep sea squids, jellyfish and marine macroalgae, all those things are this much 1.8 million metric ton is the total potential of only non-conventional, which are totally untapped in the deeper water. These are the, as I told you, myctophytes. These are significant biomass. These are mesopelagic fishes like myctophish represent one of the largest unexploited or untapped resources. And biomass of 10 to 15 billion globally it is there. Industrial promises, while not typically advised for direct human consumption because of little wax content is there. They have very good lipid protein. They have, lipid protein, and they have also little bit wax content in their body. So human, you may not directly consume it. In India, I am telling. Other countries, they are slowly with a lot of modification, lot of some research, they are now consuming. But our people, they are neither consuming nor catching. Simply they are kept on tap. And myctophytes mature at a very small size. Small size means their growth is slow, so they cannot grow very fast to a larger size. But they used to mature in the smaller size. Habitat and abundance, already those things I told. Globally, 235 species of 35 genera are recorded worldwide. Whereas in Indian Ocean, it is 137 species of myctophytes are recorded. This potential biomass is estimated to be 1 million metric tons. And as I told, this vast resource remains largely untapped due to limited direct human consumption, primarily because of their high wax content in their body. Diverse application, myctophytes are increasingly considered for industrial uses, including fish meal, nutraceuticals, oils and cosmeceuticals, opening a new avenue for resource utilization. These are the type of gear and method being adopted for catching the myctophytes. Usually this type of mesopelagic resources are having dual our migration pattern. During daytime, they will go deeper. But in the night time, they will come to the upper layer of the sea. So usually this type of fishing is done during night time. Whenever they come, actually they used to migrate between 5 to 400 meter depth during night time. Whereas during daytime, they will go beyond 1000 meter. So it will be very difficult to catch them during night time, daytime. Whereas it is easy to catch them during night time because they come upper. So we can operate the net and we can catch the fish. This is also one type of squids. As I told, non-conventional resources like our myctophids, squids also, this is there, purple back flying squid. Okay, this is also plenty in number. Around 63 lakhs tons are there lying, which is untapped. Nobody has explored these things. So these are all the methods of catching the squids, which our vessels also, we have a squid jigger to catch the ships. These are also type of fishing practices. Tuna long lining to catch the tuna fish. Have you seen? Tuna. And sailfish, marlin, swordfish. So these are the very large pelagics, oceanic resources. Very tasty and having high demand in Japan, Indonesia, Thailand, all those areas. 1 kg of fish, it goes, or 20 kg fish, it goes up to $600 to $800 per fish. But we need to have good post-harvest technology. Then only we can maintain the quality what they demand from us. So these are all for, see, these are the tunas. I am showing you the photo here. These are tunas, sailfish, shark. These all are called as oceanic resources. Okay, deep sea resources, large pelagic resources. They are large pelagic. For catching such fishes, this type of fishing methods have been deployed by Fisheries Survey of India. So what are the opportunities for deep sea resources? The first is vast unexplored biomass. Opportunities are there, lot of biomass, lot of resources are available. Only thing is that we have to go and catch it. And the second one is mesopelagic fish as future protein source. As I told you, they are having high quantity of from 11 to 13% of high lipid proteins. So they may be the protein supplement in the coming days. Scope for fisheries diversification. Scope for means everybody is catching in the coastal water. If we need to have, or if you need to explore the deep sea resources, we need to diversify our fishing methods. From near shore, we have to go to the deeper water. Whatever things needed, we have to diversify. For that, Government of India is giving lot of subsidy. Okay. And the next is nutraceuticals and fish meal industries also can use these resources. We may not directly consume it, but it can be used so that it can contribute to the blue economy of our country. And it can generate employment for the people. And technological advances also one type of opportunity. Improved deep sea fishing vessels. We have still, government is trying to just increase the number by giving lot of subsidy to the fishermen for diversification of their fishing vessels. Advanced navigation and sonar system. Government is now, our country is very much well versed with different advanced technologies. Like sonar means to locate the seas, means the fish. And other navigational equipments are also plenty. Selective and eco-friendly gears. Eco-friendly. Whatever gear means it may be a net, may be a line, whatever it may be. To catch the fish, what is used, that is called as gear. So whatever gear you are using, ensure that it is eco-friendly. It is not going to damage the ecosystem. And onboard processing and cold chain development. As I told you, infrastructure need to be developed in our countries, though it is happening nowadays, but slowly it will improve. These are resource potential. UNCLOS, I don't know how many of 2 minutes it will take. Almost it is over. Okay, United Nations, what is this? UNCLOS, Convention on Law of the Sea. So, under which every country has the right to do any kind of activity in the EZ. And government support for deep sea fishing, as I told you, scope for public-private partnership, PPP mode also, PPP mode also it can be done, and international collaboration and research also can lead to just improve our technology further. These are all challenges: slow growth and late maturity of species, low reproductive rates, high vulnerability to overfishing because they grow very slow. So anybody, if you operate any shrimp trawl, deep sea shrimp trawl, obviously this also our deep sea resources like microfids will come as bycatch. Bycatch means the catch which is not targeted, which is not useful. By this also, these are all challenges. We have to see that those things are not caught. Then limited scientific stock assessment. Stock assessment has not been properly made for deep sea resources because of lack of survey and catching, means landing of deep sea resources. Next is technological and economic barriers. What are the things? High capital and operating cost. Because once you want to go to the deeper sea, you need to have more money because more oil consumption will be there. So definitely it will be little cost effective. Requirement, skilled manpower. Simply going to the deep sea, it will not serve our purpose. You need to have a skilled persons to operate such type of things. Limited access to suitable vessel. India doesn't have much deep sea going vessels. Now government is promoting to have more and more deep sea going vessels. And uncertain economic return, because you don't know. Once you go to a deep ocean, means deeper ocean, sometimes you may not get any catch. So you are going and coming, whatever oil you consumed, that you may not get also. The money you may not get. So money realization also is a question mark. So these are the thing IOU, actually lot of things I thought of, but already I got the signal. It is too late for lunch. Everybody is hungry. If I further delay, then everybody will get angry on me. It is better before you get angry, I should stop my just last 2-3 lack of comprehensive regulations. And these are the sustainability concern, risk of resource depletion, need for precautionary approach, science-based catch limits, and protection of vulnerable marine ecosystem. All those things need to be considered for sustainability. And these are the key takeaways: untapped wealth in India's deep sea. Already those things I have covered, so I don't want to repeat again. And these are the conclusion: charting a sustainable future for deep sea fisheries. So significant opportunities, complex challenges, sustainable exploitation, balanced approach. That's all. Hope I did not bore any of you. So thank you so much for your patience. So again, before I conclude, I must say thank you to the organizing secretary and the entire people who are involved. I can understand their pain and the tension they usually carry in their head. So Dinesh sir, thank you so much. All madams, whoever have contributed for the successful completion of this conference. So thank you very much. So thank you. Jai Hind.
Thank you very much, sir, for your valuable speech and sharing your knowledge, research perspectives, and the practical understanding of the field have greatly benefited to all the participants. We are truly grateful for your time. Thank you, sir.
Lunch has been arranged in the dining hall. We kindly request all the participants to take your lunch and be seated back at 2:45 p.m.
build an AI model, but deploying it feels like a nightmare. MLOps makes this possible. You can automate
அன்பு தம்பிகளா தங்கை தம்பி மாடு பயிற்சி நம்ம
அன்பீங்களா? நீங்க மாடு பிடிச்ச அனுபவம் எதுவும் இருக்கானா?
இருக்கு தம்பி, ரெண்டுமே இருக்கு தம்பி.
நீங்க
நம்ம
அன்பீங்களா? நீங்க மாடு பிடிச்ச
அவங்க பயிற்சி நிறைய கொடுப்பாங்க.
அப்படியே கட்டி
அதாவது இன்னைக்கு இந்த நிகழ்வு எப்படி வந்ததுன்றதை உங்களுக்கு ஒரே ஒரு நிமிஷத்தில் சொல்லிடுறேன். ஆக்சுவலாக இந்த ப்ரோக்ராம் நம்ம நிமிஷத்தில் சொல்லிடுறேன். இன்னைக்கு இந்த நிகழ்வு எப்படி வந்ததுன்றதை உங்களுக்கு ஒரே ஒரு நிமிஷத்தில் சொல்லிடுறேன். ஆக்சுவலாக இந்த ப்ரோக்ராம் நம்ம நிமிஷத்தில் சொல்லிடுறேன். ஆக்சுவலாக இந்த ப்ரோக்ராம் நம்ம கல்ச்சுரல்
வித்யாரத்னா ப்ரொஃபசர் டாக்டர் எம் கருணா
ஆசிரியர்களும் மிகப்பெரிய காரணம். வணக்கம்.
Thank you, sir, for your valuable insights.
உங்களுக்கு ஸ்டாஃப்ஸை நிக்க வைக்கணும்னு நினைக்கிறீங்களான்னு எனக்கு ஒன்றும் புரிய மாட்டேங்குது.
CSI Metric Higher Secondary School. Thank you.
See, this artificial intelligence.
Mrs. Akila, History and Geography.
மீட்டிங்கோட தலைவர் யாருன்னா பாம்பேல
பண்ண தப்பு வந்து
1885 ஃபர்ஸ்ட் செஷன்
I hear 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 I hear
Research scholars, students, and all participants to the valedictory function of the National Conference on Marine Resource Conservation and Valorization of Blue Revolution. Thank you all for being a part of this meaningful, memorable occasion. Research scholars, students, and all participants to the valedictory function of the National Conference on Marine Resource Conservation and Valorization of Blue Revolution. Thank you all for being a part of this meaningful, memorable occasion. Research scholars, students, and all participants to the valedictory function of the National Conference on Marine Resource Conservation and Valorization of Blue Revolution. Thank you all for being a part of this meaningful, memorable occasion. Research scholars, students, and all participants to the valedictory function of the National Conference on Marine Resource Conservation and Valorization of Blue Revolution. Thank you all for being a part of this meaningful, memorable occasion. For women and the Department of Microbiology scholars, gives us immense pleasure to the valedictory function of the national dignitaries, honorable guests, faculty members, research scholars, students, and all the participants. Thank you all for being a part of the national conference on marine resource conservation and valorization of Blue Revolution. For women and the Department of Microbiology scholars, gives us immense pleasure to the valedictory function of the national conference, honorable guests, faculty members, for the research scholars, students, and all the participants as part of the national conference on marine resource conservation and valorization of Blue Revolution. For women and the Department of Microbiology humbly refuse to deliver the welcome address and bless us gathering with the words of wisdom. Honorable guests, faculty members, for the research scholars, students, and all the participants as part of the national conference on marine resource conservation and valorization of Blue Revolution. For women and the Department of Microbiology humbly refuse to deliver the welcome address and bless us gathering with the words of wisdom. Honorable guests, faculty members, for the research scholars, students, and all the participants as part of the national conference on marine resource conservation.
Faculties, vice principals, scientists from various research centers, research scholars, students, participants, and my dear student friends of VIAS. Good evening to all of you. Indeed, I feel great pride to welcome you all for this 2 days national conference organized by the Department of Microbiology, Sankagiri, Vivekanandha Arts and Science College for Women. And I hope you all have enjoyed this day with different amount of knowledge for this 2 days national conference organized by the Department of invited microbiology, Sankagiri, Vivekanandha Arts and Science College for Women, India, and from different research centers. Almost all our chief guests are in the scientists grade. They have oriented these 2 days to all the young minds on the importance of marine resources and Blue Revolution. We have just interacted about the theme of the conference, why we need this conference now. Yes, this is the need of the hour because the coastal areas, including the marine sources, are getting destroyed by us actually, not because of any other animals and other climatic conditions. It is being destroyed by the people, human being actually. So I think this 2 days might be a meaningful one to all the future scientists who are sitting in front of me in the form of microbiology, biotechnology, forensic science from different parts of India, especially the students from the Chhattisgarh. Almost 30 plus students have come from Chhattisgarh. Dear students, I really express my sincere thanks to all your presence today, because you have enjoyed your 2 days here. And thank you very much for accepting your invitation. I think, hope you will be here for another 3-4 years in Vivekananda Arts and Science College as the research scholars or PG graduates or what not, maybe as the professors, maybe in future. And of course, we have good number of students from the Ramnath district under the headship of Vice Principal Madam. Thank you very much, Madam, for being the part of the conference organized by our department, Microbiology. And almost we have students from other parts of Tamil Nadu, few students from Savita University, few scholars from Amrita Institute, and almost SRM colleges and other institutions across Tamil Nadu. And I extend my hearty thanks and hearty welcome to all for this valedictory session. So this is not just the beginning. This is maybe a beginning, not going to be an end. Maybe we are in a position to give the valedictory session at this 2 days national conference organized by the Department of Microbiology, but still we have just seeded a seed here. This will grow with all your support as the microbiologist. This will grow further, maybe in the future course time. Almost we are set ready to face the next conference in the same venue on February 12. The Department of Biotechnology under the leadership of Dr. Prabhakaran, we have planned a conference on sustainable development goal, but maybe yet another topic which will be meaningful to all the scientists who are sitting here. So at this moment, once again, I don't want to take much of your time, because you have to plan your travel and other valedictory process. And thank you very much for the opportunity. Once again, I extend my warm welcome to all the distinguished people who are sitting here on the stage and off the stage. On behalf of Vivekananda Arts and Science College, Sankagiri, with the blessings of our founder chairman and secretary sir, Dr. M. Karunanidhi sir, and all the management members, as a principal of this institution, once again, it is my duty to welcome you all for this valedictory session of this 2 days conference, national level conference sponsored by ANRF and CSIR. Thank you very much for the opportunity.
Thank you for being an integral part of this meaningful journey, sir. With immense respect and admiration, we extend a warm and gracious welcome to Dr. M. Palanisamy sir, Scientist E, Botanical Survey of India, Government of India, Kolkata, to grace this dignified assembly. We humbly and respectfully invite you to deliver the valedictory address, sir.
Good evening to you and all. So you are all enjoyed the last 2 days on marine resources. Without marine resources, we are not unable to survive at this age. I think, knowing or unknowingly, daily you are having some marine resource products. For example, when you are going for good functions, without ice cream you will not come out. Then ice cream, how they are making, you may be knowing. Without marine product, ice cream is nothing. You will not get ice cream. For example, carrageenan, we are extracting from red seaweeds, Gracilaria or Hypnea. From that we are extracting carrageenan. Through carrageenan only we are getting solidified ice creams. So without that you will not get it. So like that, the peoples, daily the coastal villages or any other peoples, without fish, when you take West Bengal or Kerala, daily in the meals, without fish they will not sufficient or they will not satisfy their food. So like that, the coastal resources are wide, so that to conserve this conference, National 2 Days Conference has started. And everyone might have given, all the scientists are knowledgeable scientists and different organizations. The organizations are well established organizations. All my friends who are given the lectures last 2 days, they are all from well organized institutions. For example, CSMCRI, CMFRI or CMLRI or Fishery Survey of India and all peoples, they are all almost they are in C only. Maximum times they are in C only. The organizing one of the person, Dr. Ajit, he served more than 35 years, almost 35 years. Maximum on the C only he is serving. So like that, all the peoples are focusing for how to conserve the sea resources, because day by day, due to the climate change, we are losing lots of species. So that how to conserve, not only that, the over exploitation or anthropogenic activities. So this and all the main causes for declining the marine resources. For that, how to conserve. Last 2 days, you might have a very good lectures and discussions, and you might have got lot of informations from our dignitaries and other talented peoples. And moreover, here I planned to give a lecture on marine macroalgae. Due to time constraint, I am unable to. Same thing without any pictures, I am showing all these things, telling all these things. For example, when you are going to the seashore, you can see the plenty of assured materials, that is flora. How many marine flowers are there? Okay. All over Indian coastal region, we are having more than 900 floras are there. Faunas are mostly high, but in flora concern, it is more than 900 species are available in our Indian coastal regions. That is called marine macroalgae, known as seaweeds. You may be having the name of seaweeds. For example, when you are simply we are telling alva. When you are going to the seashore area, green talus will be there, that is called alva. When you are going little bit interior, you can see the grapes like suchars, that is kalarpa. So you might have heard the names kalarpa or alva. These are green algae. When you are going deep, you can see the plenty of red algae and brown algae, that is called Gracilaria, that is red algae. When you are going for a deeper, then you can see the floating or in deeper sargassum in brown in color. So that and all having the plenty of potentials. Why just I am telling means that having a lot of potentials. When you are going to the pharmaceutical industries without marine macroalgal products, it is very difficult to complete your research. And when you are going to the biotechnology, what is the importance? Agar-agar media. So agar-agar media from which we are getting, that is agar-agar powder, from red seaweeds. You know, no? Jadilla acidosa, that is a species. All over the world we are getting #1 quality agar-agar. And Gracilaria also, Edilis is there, but compared to Edilis, Jadilla acidosa giving the #1 quality agar-agar. So this one I am planning to give in my lecture, but unable to, so that means time concern, I am unable to give that one. And last 2 days, the institution, Vivekanandha institution given a very good platform to the young budding researchers and students. They might have learned all the marine bioresources and how they are utilizing and how they are monitoring the marine bioresources with remote sensing, and how they are cultivating whether it is plant or animal, whatever it may be, how they are culturing or cultivating, and how we can utilize the marine resources for our medical or our health or farming or your pharmaceutical companies or biofuel and all other purpose, how we can utilize. You might have heard or you might have learned all these things. And this program, I hope, without the institution, this Vivekanandha institution, you will not have this function would not be success, because wherever you go, there the places are very, very less. You see the auditorium, it is a big, very wide area. I think at a time we can 1,000 members we can sit, I think. Nowhere I have not seen so far this kind of auditorium, because in BSI or JSI or the Ministry of Environment and Forestry, we used to arrange our national conference or international conference, but it will not be like this. But we used to very small area only. But here you see it is a very big area, and we have to thanks to Vivekananda educational institutions, because earlier I had been to this one more Elayambalayam. So Elayambalayam also. They are also very big institution. That means the same organization. They are having they are also one center, So there also I have given, but there also they have maintained very well, and here also the campus and the institutions and all the departments are well established, and they are very nicely or neatly they are managing. The conference itself, the conference hall itself, we can say how the institution, they are teaching to the students and how the facilities they are created. So really excellent. And the organizing committee chairman and the other co-chair and other peoples, they have very nicely arranged this program. And once again, I am thanks to one and all for on the dais and all our young budding researchers. Thanks to one and all for this attending this well-organized program. Thanks to one and all. Thank you.
Thank you, sir.
Now, with immense respect and gratitude, we take great pleasure in honoring and welcoming our distinguished guest. Now, I request Dr. V. Padmanabhan, sir, Principal of Vivekanandha Arts and Science College for Women, to honor Dr. D. Dinesh Santal Lejoni Maurabain, sir, Principal, Scientific Officer, Rajiv Gandhi Center for Aquaculture, Ministry of Commerce and Industry, Kerala. Thank you, sir. Next, Dr. K. Anandaraja, sir, Senior Scientist, ICAR, Central Institute of Brackish Water, Aquaculture, Ministry of Agriculture, Chennai. Next, Dr. K. Anandaraja, sir, Senior Scientist, ICAR, Central Institute of Brackish Water, Aquaculture, Ministry of Agriculture and Farmers Welfare, Chennai. Next, I request Dr. B.T. Suresh Kumar, sir, Director IQAC, to honor Dr. Sujit Kumar Patnaik, sir, Junior Fisheries Scientist, Ministry of Fisheries, Animal Husbandry and Dairying, Government of India, Mumbai. Next, Dr. M. Palanisamy, sir, Scientist E, Botanical Survey of India, Government of India, Kolkata. Thank you, sir. True guidance creates leaders, nurtures talent, and leaves a lasting legacy. We proudly welcome Dr. B.T. Suresh Kumar, sir, Director IQAC, Vivekanandha Arts and Science College for Women, to felicitate the address. With immense respect, we welcome Dr. R. Dinesh Kumar, sir, Assistant Professor, PG and Research Department of Microbiology, to present the conference report.
So very good evening to all. So I am Dr. Dinesh Kumar, Organizing Secretary, Assistant Professor, Department of Microbiology. I am very happy to and proud to read the report of our last 2 days National Conference on Marine Resource Conservation and Valorization for Blue Revolution, MRVB 2026, which is sponsored by ANRF, CSIR and CMLRI, Kochi. In collaboration with ICR, NGFERA, Kerala. In our conference, totally 1,032 participants registered through online and offline mode. It is a first record in our VA campus. 1,032 participants participated from 72 central and state university and institution different colleges, including scientists, research scholar, faculty members, students and other participants. Like Kalinga University, Raipur, Chhattisgarh, the American College, Madurai, Muhammad Sada College, Arts and Science College, Ramanathapuram, Central Fisheries Research Institute, Cochin, Kamadana Fisheries University, Gujarat, and Satyabama University, ICR NGFR Kochi, Dr. MGR University, DNJFU Tamil Nadu Fisheries University, Thoothukudi and Chennai and Poneri campus, Kerala University, Government College, Bharatiyar University, Bharathidasan University, Annamalai University, SRM, Savita University, National College Trichy, Madurai Kamarajar University, Manomaniyam Sundarnar University, Dr. NGP College, Krishnamal College, BSc Arts and Science College, Coimbatore, Karpam University, Coimbatore, and our participant also. Totally we are received 352 oral abstract, 47 poster abstract received our conference. So we are planned to publish after our conference, selected full length papers published in essay journals, our conference publication partner, Springer Nature and elsewhere, like environmental monitoring assessment, impact factor 3.1 is a essay indexed journal. So another one journal is environmental pollution and research publication partner, Springer and Nature, impact factor 5.1, and food and humanities essay journals publication partner elsewhere, impact factor 1.2. I am very happy, gratitude for the opportunity. My special thank to our chairman secretary sir and our management, and I special thank to our lovable principal Dr. P. Batman sir, and my heartbeat thank to our workaholic dedicated our director IQC sir, thank you, and management, our faculty members. Particularly I thank to our second MSc microbiology student, third BSc microbiology and first MSc microbiology, and all first year and first UG and first PG student, and special thank to our Kausalya madam, our team members, and Sonia Sri also. Thank you.
We shall announce the prize winners. I would like to invite Dr. R. Maithili ma'am, Head Department of Microbiology, to announce the prize winners.
Dr. Prince Pragas Jabba Kumar, Scientist E, Coastal and Environmental Engineering, NIOT Campus, Chennai. He received Best Scientist Award. Welcome you, sir. So next, I welcome Dr. Raja Kaliyappan sir, co-founder and director, Dogma Center for Life Science. So he received the Best Entrepreneur Award. Congratulations, sir. So next I welcome Dr. R. Manohar Jabakumar sir. So co-founder, head. Dogma, sir. What's Dogma? So Manohar Jabakumar sir. Next, Dr. S. Selvi ma'am, Associate Professor, Bharatidasan College of Arts and Science. She received Mid-Career Women Researcher Award in the year 2026. Welcome you, ma'am. So next I call upon Dr. Anita ma'am, Assistant Professor, AIMT University. So she received Young Researcher Award. Congratulations, ma'am. Welcome you, ma'am. Next, I call upon Dr. V. Priyalakshmi, Assistant Professor, Muhammad Sadaq Hamid College of Arts and Science for Women, Ramnath. She received Young Women Microbiologist Award in the year 2026. Welcome you, ma'am. Next, I call upon Dr. J. Renuka ma'am, Assistant Professor, the American College, Madurai. She received Emerging Women Microbiologist Award in the year 2026. Welcome you, ma'am. So next, I call upon Dr. S.R. Sivakumar sir, Assistant Professor, Bharatidasan University, Tiruchirappalli. He received a Research Excellence Award in the year 2026. Congratulations, sir. Welcome you, sir. So next I call upon Dr. R. Preeti Ratna ma'am, Assistant Professor, Hindustan College of Arts and Science, Chennai. She received Early Career Women's Researcher Award in the year 2026. Welcome you, ma'am. So next I call upon Dr. E. Suresh sir, Assistant Professor, Tamil Nadu Dr. J. Jayalalitha Fisheries University, Chennai. He received Senior Scientist Recognition Award in the year 2026. Welcome you, sir. Next I call upon Mr. T. Muthurayar, Assistant Professor, NMSSVN College, Madurai. He received Microbiology Research Excellent Award in the year 2026. Welcome you, sir. Next, I call upon Ms. S. Sri Ranjini, Research Scholar, Shastra Deemed University, Kumbakonam. She received a Yearly Career Women Researcher Award in the year 2026. Congratulations, ma'am. Welcome you, ma'am. So next, I call upon Ms. V. Ramabai, Research Scholar, AIMT Deemed University. She received a Rising Women Scholar Award in the year 2026. Welcome you, ma'am. So next I call upon Ms. Maria Susila Vinisha, Research Scholar, Tamil Nadu Dr. J. Jayalalitha Fisheries University, Chennai. She received Women Research Award in Fish Genetics in the year 2026. Welcome you, ma'am. Next, I call upon Ms. S. Indumadi, Research Scholar, KSR College of Arts and Science for Women, Tiruchangod. She received Rising Women Microbiologist Award. Congratulations, ma'am. Welcome you, ma'am. So next I am call upon Mr. B. Vinayagamurthy, Research Scholar, Savitha University, Chennai. He received a Rising Scholar Award in the year 2026. Congratulations, sir. Sir Vinayagamurthy, sir. Next, I call upon Mr. Rahul Kasi, Research Scholar, Amit University, Chennai. He received a Rising Research Scholar Award in the area of marine biotechnology in the year 2026. Welcome you, sir. So next, I call upon Mr. K. Poolpandi, Research Scholar, Kamaraj College, Thoothukudi. He received a Early Career Researcher Award in the year 2026. Welcome you, sir. Next, I call upon Mr. Hariharan P., PG Research Scholar, Tamil Nadu Dr. J. Jayalalitha Fisheries University, Chennai. He received Best Thesis Award in the discipline of MFSC. Congratulations, sir. Mr. Hariharan, sir. On behalf of organizing committee, I once again congratulations to all awardees. Congratulations to all. So next, I would like to call participants and present a certificate from various colleges and university. Based on the time construction, I call upon one person from each university to receive the certificates. The American College, Madurai. So any one person can receive the certificates. The American College, Madurai. To represent any one person from the American College, Madurai. So ICAR, NBFGR, Kerala, Cochin. So CSR Institute, ICAR, NBFGR, Kerala. Okay, sir. 2 people can come to receive the certificates. From each institution, 2 people can receive the certificates. Annamalai University. So next I call upon Annamalai University. Next I call upon Mohammed Sadak, Hamid Arts and Science College for Women, Ramanathapuram. Mohammed Sadak, Hamid Arts and Science College for Women, Ramanathapuram. K.S. Rangasamy College of Arts and Science, Autonomous, Tiruchangod. Dr. MGR University. So next I call upon Dr. MGR University students. So next I call upon ICR Institute, that is Central Institute of Fisheries, Mumbai. from Mumbai. So next I call upon Tamil Nadu, Dr. J. Jayalalitha Fisheries University, Chennai. Next I call upon Kerala University. Next I call upon Kerala University participants. Next I call upon JKK Government College, Kumarapalayam. Next I call upon Vidya Arts and Science College. So next I call upon Bharatidasan Arts and Science College, E-Road. Next I call upon Bharatiyar University, Coimbatore. So next I call upon Bharatiyar University, Coimbatore. Next I call upon Karunya Institute of Technology of Science. Next I call upon Karunya University. Next I call upon National College, Trichy. Next I call upon National College, Trichy. Next I call upon Kamraj College. So next I call upon Excel College, Excel Polytechnic College. Next I call upon Savya Banu Sastriya College. Savya Banu Sastriya College. So next I call upon Ratnam Arts and Science College. Next I call upon Ratnam Arts and Science College. So next I call upon Dr. NGP Arts and Science College, Coimbatore. Next I call upon Dr. NGP Arts and Science College, Coimbatore. Next I call upon Sona College, Salem. Next I call upon Sona College, Salem. Next I call upon Satyabama University, Chennai. Next I call upon Satyabama University, Chennai. Next I call upon Ethiraj College, Chennai. Next I call upon Ethiraj College participants from Chennai. So next I call upon PSG Arts and Science College, Coimbatore. Next I call upon PSG Arts and Science College, Coimbatore. Next I call upon Alagappa University. Alagappa University from Karaikudi. இது இல்லாதது யார்? So next I call upon Union Christian College. Union Christian College. So next I call upon Das BV College. Rameshwaram. So all students are come to receive the certificates. Muttayamal Arts and Science College from Mrcvbr. Next Kerala University. Illana. Satip. So Muttayamal Arts and Science College. So next Muttayamal Arts and Science College, Mrcvbr. Next Padmavani Arts and Science College, Salem. Muhammad Sada College participants all are invited to stage to receive the certificates. Please come on to the stage.
Muhammad Sadak Arts and Science College for Women, Ramanathapuram, please on the stage.
Next I call upon Kalinga University, Chhattisgarh, Raipur. Next Savita University, Chennai. Savita University participants from Chennai, come and receive your certificates. Please come on to the stage. Manonmaniyam Sundarnar University from Tirunelveli. Manonmaniyam Sundarnar University from Tirunelveli. Manonmaniyam Sundarnar University, Tirunelveli. Kailash Women's College. Kailas Women's College, Karnataka College, Karnataka College, Magalashmi Women's College, Magalashmi Women's College, Amrit University, Amrit University, Sri Ramakrishna Mission Vidyalaya College of Arts and Science, Coimbatore. Amat University, Amat University, Chennai. So Manonmaniyam Sundarnar College, Tirunelveli. Sri Ramakrishna Mission Vidyalaya College of Arts and Science, Coimbatore. Ramakrishna College, Coimbatore. Government of Arts and Science College, Salem. Magalashmi Women's College. Kalinga University, Chhattisgarh, Raipur. All participants come onto the stage. Thank you, sir. Kalpana Krishna College. Ma'am, மூணு மட்டும் சொல்லுங்க மேம். சர்டிஃபிகேட் கொடுத்து சும்மா வாங்கிக்கோங்க. ஹரியா ஃபர்ஸ்ட். ஹரியா தாசனி. அல்ஜர்சனா தாசனி காலேஜ். கல்பனா கிருஷ்ண காலிங்கா காலேஜ். நான் வாக்கிட்ட. இது என்னது? ஃபரியா. ஃபரியா இது அல்ஜர்சனா. போஸ்டர் ரீட் பண்ணுங்க. ஃபர்ஸ்ட் செகண்ட் தேர்ட் அது போஸ்டர்ல அவார்ட் தனியா மூணு அவார்ட் பண்ணிடலாம். ஃபர்ஸ்ட் பிரைஸ் ஓரல் முதல்ல இது சொல்றா. ஃபர்ஸ்ட் செகண்ட் தேர்ட் ஓரல்ல ஃபர்ஸ்ட் செகண்ட் தேர்ட் அடுத்த அவார்ட்ல அஜித் குமார் சம்பத்குமார் கடைசி சேர்மன் சார் அவார்ட். ஓகே போஸ்டர் இதுமா போஸ்டர் ஓரல் அவார்ட் அவ்வளவுதானா? போஸ்டர் ஓரல் அவார்ட் அவார்ட்ல ஐ அம் கிளியர். அஜித் குமார் ஃபர்ஸ்ட் சம்பத்குமார் ஃபர்ஸ்ட் கடைசி சேர்மன் சார் ஓரல் ஃபர்ஸ்ட் பேர் ஒரே மாதிரி. அது ஸ்டார்ட் அப் வந்து ஸ்டார்ட் அப் கொடுக்கல. ஸ்டார்ட் அப் கொடுத்து இல்ல ஸ்டார்ட் அப் கொடுத்து பேசுங்க. பரவாயில்ல ஸ்டார்ட் அப் எழுதுங்க. ஸ்டார்ட் அப் சர்டிஃபிகேட் மட்டும் நம்ம பேஸ் எது இல்லைமா? ஓகே மூணே பேர் தான் அப்படியே பண்ணிருங்க. இது ஸ்டார்ட் அப் ஃபர்ஸ்ட் யார்? ஃபர்ஸ்ட் இவங்க ஸ்ரீநந்தி மாலி நாமளே நியூஸ் இதுனா கண்டுபிடிச்சீங்களா? உங்க குடுமா ஃபர்ஸ்ட் ஸ்ரீநந்திங்க. சிஏஎஸ் இன் மேரின் பயர் சிஏஎஸ் இன் மேரின் அண்ணாமலை யூனிவர்சிட்டி. உங்க ஸ்காலர் செகண்ட் இயர். Next, I proudly call upon prize winners for startup competition, national level startup and display competition in the year 2026. In collaborated with ICAR, NBFCR, Kerala, Kochi. So the first prize goes to Srinidhi Himani. Srinidhi Himani from Annamalai University. CAs in Marine Biology. The second prize goes to P. Sivanation. The second prize goes to P. Srinivasan. The third prize goes to Anbu Selvan. The second prize goes to P. Sivanation from Bardiyar University, Coimbatore. Anbu Selvan. The third prize goes to S. Anbu Selvan, Annamalai University, Marine Biology. So, congratulate to all startup winners. So, next I proudly read prize winners in poster presentation in our national conference. The third prize goes to Kalpana and Krish from Kalinga University, Chhattisgarh. The third prize goes to Kalpana and Krish. Both of you win the third prize. Congratulations. So the second prize goes to Ajarshana from Dasami College. Ajarshana from Dasami College. Next, the first prize goes to Miss Faria from Dasami College. The first prize goes to Faria from Dasami College. Once again, I congratulate to all poster winners. So second prize goes to Ajal Sarshana, Dasami College. Aljasana from Dasami College. The first prize goes to Faria from the same college, Dasami College. So in oral presentation, the third prize goes to Noorul Aishan and Janadul Firdosha from Muhammad Sada College. The second prize goes to Nereen and Fegadul from ICAR NBFCR, Kerala, Kochi. Second prize goes to Nerin and Fragadul from ICR NBFGR, Kerala, Kochi. So the first prize goes to Sugos M from ICR CAFE, Mumbai. The first prize goes to Sugos ICR CAFE, Mumbai. Next I read Best Oral and Overall Poster and Oral Presentation Awardees. Dr. T.T. Ajit Kumar Sir Award received by Harish Kant from Annamalai University. Congratulations, Harish Kant. So next, the second prize, next, the Sambat Sir Award goes to Srinidhi Himani, Annamalai University. Second prize goes to, Sambat Kumar Sir Award goes to Sri Nandini Himani from Annamalai University. Congratulations, ma'am.
The next, another one important moment, the next award, overall performers, our marine conservation and blue economy. This is one of the important award in the year of 2026. This award name is Professor Dr. M. Karnanidhi Sir, Chairman and Secretary. Award goes to P. Soundarya, Periyar University, Salem. One of the important awards. Professor M. Karnanidhi Sir, Chairman and Secretary. Best Performer Award of our conference, P. Soundarya, Periyar University, Salem. Periyar University, Department of Microbiology, Salem.
We sincerely thank our respected dignitaries for gracing this prize distribution ceremony. With great respect and pleasure, we warmly welcome Dr. R. Maithili ma'am to deliver the vote of thanks.
On behalf of our organizing committee, so I proudly feel to thank all the participants. First of all, I would like to thank our most respected chairman and secretary, sir, to giving this wonderful opportunity. And also I thank our management peoples. Next, I would like to thank our most respected dignitaries on the stage and off the stage. So next, I would like to thank our beloved principal, sir, Dr. V. Padmanathan, sir. His continuous encouragement and support only we successfully complete this conference. Thank you, sir. So next, I would like to thank our directors, IQSC director, sir, Dr. P.T. Suresh Kumar, sir. His encouragement and continuous motivation only we successfully complete this conference. Thank you very much, sir. So next, I would like to thank Organizing Secretary Dr. R. Dinesh Kumar, sir. Next, I would like to thank our organizing committee team members, and I also thank HODs from various departments, faculty members, and also participants, that is, which include scientists, researchers, faculty members, and scholars from various institutions and university. So, thank you to your cooperation and continuous support these 2 days, and also thank media, press people for this backbone of this conference. So once again, I thank you to all. Thank you for this opportunity.
So, just wait a minute. It is our humble request that if you can share your feedback about this 2 days conferences, so you can at least one representation from one college can come and share your feedback, suppose if you want to share. Come to the stage. Can you please come to the stage? Suppose if anyone want to share. Already you have shared your feedback yesterday, but still we got to know that few of your students would like to share your feedback. Yes, that is why we are calling you. Yes, thank you, madam. And if anybody is willing to share your feedback, you please be ready. Yes, please come forward come forward to the stage. Please come to the stage. Thank you. So one person can represent your institution.
Good evening, everyone present over here on the dais and off the dais. So it's my pleasure to give me the feedback on behalf of Tasim Bibi Abdul Qadir College. I'm Dr. Aisha, Vice Principal of that college. It is, see what I could tell you, from yesterday morning 2:30 onwards, we have been engaged with all the faculty members, especially Dinesh Kumar sir and the students of microbiology department. And I should especially thank this management for providing us a very good platform to get knowledge on this blue revolution. You can give applause now. And I should thank my principal also, Dr. Sumaya, for giving us this opportunity to attend this wonderful program. And I sincerely extend my sincere thanks to, the hospitality and all other arrangements being made for each and every one of this conference. And நிறைய பேசணும் எதுன்னு மறந்துருச்சு திடீர்னு கூப்பிட்டாங்களா ஒன்றும் பேச தெரியல சரியா okay so and then moreover you people have talked only about the hospitality and other things. Okay, it is okay, but the quality of the conference, no one has talked about the quality of the conference. And I should thank the associated partners also in this occasion. And the blue revolution is one of the most important aspects in which we should see many things. And I especially thank the associated partners of the conference and the microbiology department, especially the head of the department and other faculty members, for giving us an enriched knowledge on this blue revolution. So thank you very much, madam. And Last but not least, I should thank the principal of this campus, who is a very dynamic personality, and he was not at all tired till 11 o'clock. I could see his enthusiasm and motivation towards us. Sir, really you are motivating everyone, sir, and all other members like IQAC coordinator and my senior, the director, sir, and he is also very pleased to invite all of us for this conference. And you know, the hospitality will be extended by only very few members, but at this juncture I want to express that every one of the higher authorities, they are ready to serve for us. So this is behind the success of this Vivekananda College of Arts and Science. I hope so. And I extend my sincere thanks to the the chairman of this esteemed institute. Since even though they are running 25 institutions, I could see the dynamism of all other leaders. So the leaders are leading this college in an excellent way. A great salute to all the leaders of this institution on my behalf. Thank you very much for everything, sir, you provided. Thank you, sir.
So I would like to invite all the microbiology faculty to the stage. Please come. All the microbiology faculties, you please come to the stage.
Thank you, sir. Microbiology students, please go and utilize our college bus immediately. Microbiology students, please go and utilize our college bus immediately.
We are students, if anybody wants to...
Our existing students, we are students, microbiology students, please go and utilize our college bus immediately.
Sir, we can have national anthem. After that, they may go now. Just wait for a few minutes, students.
My students, please wait for here.
Just wait for a few minutes. Yes, we'll start national
Just
anthem.
a few minutes, please. Sorry for the inconvenience.
Microbiology students, you please wait. The bus will wait for you. So at this moment, let us all give the biggest round of applause to the team microbiology. That is why I have called all the people, faculty here. Yes, Pandian sir, come to the stage. So this is the team microbiology headed by Dr. Maithili. Yes, please come, sir. And Professor Dinesh, Professor Pandian, Professor Sonia Sri, and Professor Kausalya. Because of this collective team effort, this conference resounds a huge success, and let us all express our sincere applause to all these people because of, and also the students of microbiology. You can give them the big round of applause to the students of microbiology, the team microbiology. And two more persons I left over here, the research scholars of microbiology, please come to the stage. The scholars, please come to the stage. Two scholars, where is he? Yes, come. They work hard to make this day a meaningful one, and on behalf of everyone sitting here and participated in this conference, we would like to extend a good salute to all the team members of the team microbiology of VIAS. Thank you very much. We may proceed with national anthem.
Participants who require corrections in their certificates are requested to collect them near the mini hall after the completion of program. It's time for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे